I also want to thank the other members of the organizing committee, Dr. Betty Kahn from Kaiser Permanente of Northern California, Nancy Davidson, my colleague here from Fred Hutch and Seattle Cancer Care Alliance, Electra Pasquette from The Ohio State University. Just a brief overview of the day of the symposium today. It's sort of a framework setting, so we're pleased to have Dr. Ned Sharpless as giving our keynote address. Then I'm going to talk about some research opportunities in the Women's Health Initiative, and then we have a great talk by Dr. Supriya Mohili, who will give more of a clinical perspective on the issues of cancer and aging. Tuesday is mostly focused on the biological aspects of aging, and we have a variety of talks around different biological mechanisms that might be want to be considered, and then starting to look more at some of the clinical phenotyping issues, which will take us more into day 3. We've had to do some adjustments with schedules. So there's a talk on immunology first thing Wednesday morning, and then nutrition and physical function and frailty on Wednesday. So that's a brief overview of where we're going. Some reminders, this session is being recorded. So if that isn't okay with you, either stay muted and your camera off, or we'll talk to you later. And please keep your mic muted and enter your questions in the chat. And when we're in the Q&A section, if the moderator calls on you and you want to mute and ask your question directly, that's fine, but please wait for the moderator to call on you. So with that, I would like to introduce Dr. Tom Lynch, the director of the Fred Hutchinson Cancer Research Center, who will introduce our keynote speaker.
Dr. Anderson, thank you so much for your for organizing this symposia and bringing such a remarkable group of speakers and investigators to the Fred Hutch. Obviously, we all wish that this was in person, and soon it will be because of the work of so many people working tirelessly on COVID. But it's great to be able to take our heads and minds and think about something that is at our heart and soul, which is cancer and the impact of aging. And it's with great pleasure that I get to introduce Ned Sharpless. Norman Sharpless was officially sworn in as the 15th director of the NCI on October 17th of 2017. Prior to this, Ned was the director of the Lineberger Comprehensive Cancer Center at UNC. Dr. Sharpless was a Moorhead Scholar at UNC Chapel Hill. He got his MD degree from UNC in 1993, and he then was recruited to the Massachusetts General Hospital, where I met Ned as an intern in 1993. I was a second year attending on the oncology service when Ned was an intern there. And he completed his residency there, then did his hematology oncology fellowship with us at the Dana-Farber Partners Fellowship at Harvard Medical School. He did his postdoc in Ron DePino's lab. And then after 2 years on the faculty at HMS, joined the faculty at University of North Carolina in 2002, becoming the Wellcome Professor of Cancer Research in 2012. Ned's a member of the Association of American Physicians and the American Society for Clinical Investigation. He's a fellow of the Academy of the American Association of Cancer Research. He's authored more than 160 original scientific papers. He's also co-founded 2 clinical stage biotechnology companies. And he served as acting commissioner of the FDA, of Food and Drugs at the US FDA for 7 months in 2019 before he returned to be the director of the NCI. I think those of us who know Ned know that he is a person of extraordinary integrity and curiosity, a spectacular scientist, a wonderful physician, someone who spans the entire gamut of what you want to see in a cancer researcher. And we are so lucky as a cancer community that he has given so much to us in his role, both at the FDA and at the NCI, made a huge difference. And so, Ned, it is just terrific to welcome you to the Fred Hutch.
Great. Well, thank you, Tom. It's wonderful to be here, virtually at least. I'm sorry we're not doing this in person because, Fred Hutch has been such an important part of giving advice to the NCI. There's so many great advisors. Gary Gilliland through the years has been extremely helpful. Nancy Davidson has done a number of jobs for the NCI, including particularly challenging with the president. And of course, Tom, you know, the little I'm going to talk about lung cancer today, and the little I know on that topic, I think I learned from Tom Lynch long ago. So, you know, and it's of course a terrifically exciting scientific milieu. So someday, as Tom pointed out, this will be over and we can travel again. And I look forward to an in-person visit if that's possible. So I thought what I'd do today is start by talking a bit about some opening remarks about the relationship between cancer and aging in general, which was a research interest of mine. Mention a little bit of research that you know that I started as an academic and have continued an interest in as a federal official, and then finish with some remarks about the NCI's portfolio with regard to cancer and aging in general that I think will be of interest. So first slide, please. But I thought I'd start out by pointing out an important anniversary. So this year is the 50th anniversary of the National Cancer Act. The NCI is really gearing up for discussing, you know, the importance of the NCA and what it's meant to cancer research all year. You can see that all these sorts of logos and things we've created are not NCI branded. This is, we envision that all stakeholders who have an interest in cancer progress and cancer research will take up this time to commemorate this important anniversary. I mean, it's not really an anniversary to celebrate. I mean, we're 50 years in and we still have 600,000 people dying a year from cancer. So we have a lot of progress yet to make in cancer. But I think it's also important to articulate to the public and to Congress how much progress we have made in the last 50 years and why we think the prospects for future cancer research are bright. And I hope that everyone involved in cancer research, and particularly the cancer centers, will sort of take up this charge and use this opportunity to speak nationally on this topic. Next slide, please. So I'd like to start the cancer and aging part by making an observation, which is that there's this strong association between cancer incidence and aging. And, you know, aging is in many ways the strongest risk factor for cancer, at least for most of the common cancers that we work on, as shown here. So these are SEER data looking at age of onset by sort of common tumor type. And you can see all these sort of adult solid tumors, as well as leukemia, have this sort of characteristic pattern. It's not really clear why the incidence of cancer goes down at above sort of age 75, whether that is an effect on screening and diagnosis, or if that's a true biological finding, but it is reproducible observation. But I think it's fair to say that something about aging it drives carcinogenesis, and that's been a topic of real scientific interest for a long, long time. Next slide, please. One point that I think has been particularly interesting is the recent finding using molecular signatures that, you know, the passage of time itself is a mutagen. And so this is a figure that I think is probably known to most of you about the sort of mutational signatures presence in each cancer type. And you can see signature 1 and signature 5 are virtually present in every kind of human cancer. That's the first and fifth line. And one can tell that those are these sort of clock mutations, the signatures that are associated with passage of time. I think there's some animation on this slide. Next slide, please, or next click. All right, so one is this C to T transversion, which is associated with cytosine deamination, and 5 is of unknown cause, but it's a common time-associated mutation found in all cancers. Next slide, please. And areas of ongoing research. And some of these mutations that are driven by the passage of time are known to be, you know, professional card-carrying oncogenic mutations. And so we know that were you never exposed to any sort of environmental mutagen or carcinogen, just the proliferation of cells and cells sitting, you know, with the passage of time will accumulate mutations that can be oncogenic. And so that's, I think, an important part of the relationship between cancer and aging, but certainly not the only one. Next slide, please. So there's a lot of ways to take this topic of cancer and aging. I kind of break them into 3. The first is this issue of how does aging lead to cancer? And there are lots of interesting topics here about telomere shortening and dysfunction and misbehavior of stem cells, particularly as they change epigenetically and genetically, we now know, and alterations of cellular immunity and changes in the extracellular milieu. And then also down at the bottom here is an important area of geriatric oncology where we know that the onset of age limits our ability to treat cancer. So certain therapies that are quite appropriate for younger patients with cancer are less effective for older patients. And why this is and how this comes about is an interesting topic. And particularly if there's anything you can do clinically about this, it's important to talk about. And then the middle one here is what I really want to focus on today, which has gotten, I think, the least attention of the 3, but I think is interesting in and of itself because it has real advantages to studying aging. And that is that in some ways, some of the cancer therapies we use, particularly cytotoxic DNA damaging therapies, are sort of an iatrogenic model for aging in humans. And there are many of the features of aging that we think of physiologic time passage induced aging are induced by DNA damaging therapies. And that includes, epigenetic exhaustion of stem cell compartments and the induction of senescent cells, which I'll talk a bit about both of those. Next slide, please. So I thought I'd focus on this middle one today. The first 1 and 3 are really interesting too, but I just can't do everything. And 2 is one where I did do some time as an academic, and I think there's news. Next slide, please. So let me just point out there are sort of 2 kinds of ways that we think that DNA damaging agents cause aging, physician-induced iatrogenic aging. One is we have pretty good evidence now that senescent cells, which we think are play some role in promoting aging, that they're induced by DNA damaging agents, and then they persist for a very long period of time, if not forever, in intact organisms. And then also we know that when particularly the myeloid, the hematopoietic compartment is kind of killed off by chemotherapy, that forces stem cells to divide to repopulate those missing hematopoietic progenitor cells. And we know that that's bad for stem cells and will kind of induce a early aging. That finding actually is pretty old. That was discovered by David Harrison in the 1970s. Before we even knew what stem cells were, we knew that one could exhaust them with serial chemotherapy. So it's an old finding and it's of great clinical relevance. Every oncologist has the experience of treating patients with cytotoxic drugs over and over again. The drugs are still working, but at some point the counts don't recover and one has to stop treatment. Next slide, please. So senescent cells, I thought I'd start with. I think this is now becoming a reasonably well-known topic. It was for many years a very controversial topic, but I think in the last few years it's really caught on, both from basic scientific understanding of the topic as well as now industry interest in both the quantification and removal of senescent cells. And suffice to say, it's still a problem telling if a cell is senescent, in fact. They make these characteristic markers, which are shown here, decreased telomeres, expression of P16. They have this beta-galactosidase activity that's been used mostly in vitro to identify them. They make a bunch of factors that are associated with inflammation and immunity, like IL-6 and IL-8 and various proteases and chemokines. And then they have a few things they don't do, like make laminin B1, and they have generally characterized by increased NF-kappa B signaling in general. And these cells are generally produced by a number of stimuli, DNA damage, but epigenetic changes and a variety of other insults. And then they don't divide and they stick around for a long time and they make humors that may be bad for you. And there's pretty strong evidence now that the accumulation of senescent cells is beneficial in some ways because that prevents one from getting cancer. This is an important anti-cancer mechanism. But on the other hand, the accumulation of these cells throughout life appears to have toxic effects that may be tractable to therapeutic approaches. Next slide, please. So I'll relay some of the evidence for that. But first, I want to point out senescent cells were discovered by Leonard Hayflick. This slide has animation, I think. So next slide, please, or next click. So Dr. Hayflick discovered this back in 1961. And this is the rejection letter he received when he tried to publish this result at the Journal of Experimental Medicine. Next click, please. And the manuscript was rejected. He claimed to identify senescent cells, which are cells that will sit in culture forever and won't divide. And the response was, well, that's nuts. Everybody knows that the largest fact to have come out of tissue culture in the last 50 years, in 1961, was that cells inherently capable of multiplying will do so indefinitely if supplied with the right milieu. Meaning the implication was that if Dr. Hayflick had just known how to wash his glassware properly, none of this artifactual senescence wouldn't have occurred. And next click, please. That interesting letter is signed by Peyton Rouse, perhaps one of the most famous scientists in the history of cancer research, a Rouse sarcoma virus, who won the Nobel Prize in 1966. I like to point out that Peyton Rouse was really the first person to really sort of envision the idea of oncogenes. It was the finding that DNA tumor viruses could cause cancer that led to the cloning of RAS and SARC and the original oncogenes. But senescence, this obvious observation of Leonard Hayflick, is really the first discovery or the first phenomena attributable to tumor suppressor genes. And for those of you who've been in basic science of cancer, you know that tumor suppressors and oncogenes have had this sort of war of primacy in the basic research field for 50 years, and the oncogene guys always seem to win. But it's starting way back then. Next slide, please. I also have to say that Dr. Hayflick is still a very vibrant individual. This is from a conference I attended about 2 years ago in Santa Barbara, where he came and asked a bunch of hard questions, and he's still really interested in aging research. But I think that senescence is a real thing. It does really happen. It's an important anti-cancer mechanism, and that's been established by fact. If you read the original paper, it says that, senescence ought to have something to do with cancer, and it does. That's true. The other thing Dr. Hayflick postulates is it ought to have something to do with aging, and that I think turns out to be true too. Next slide. So I'll just state the theory very precisely here. That is that senescent cells accumulate at a rate that increases rapidly with aging. And this is good because it prevents those cells that are damaged from becoming cancer. It also has some role in wound healing, and it may play some role in limitation of atherosclerotic plaques. There's some benefits to senescence, but it's also bad in that these cells, they don't do what they used to do. They occupy key cellular niches. They elaborate these toxic cytokines. We think they can cause anatomic obstruction, and they don't, if you had a lymphocyte that used to, divide and fight tuberculosis or something, and that cell becomes senescent, it can't do that replicative function anymore. So it may not be able to perform its function if that function requires cellular division. Next slide, please. So if this model were right, it makes three predictions. One is that you ought to be able to tell, people ought to age differently based on their set point for senescence. There ought to be polymorphisms of the senescence machinery that leads to different aging phenotypes. That turns out to be true. Molecular aging should be measurable. You ought to be able to calculate how many senescent cells someone has and decide sort of in molecular terms how old they are. That may be true. That's an area of intense research interest, and I'll talk about NIH plans in this topic later on. And then lastly, and perhaps most provocatively, is the idea that if you had senescent cells or you got rid of them somehow, using a senolytic, a small molecule inhibitor, an antibody, maybe a CAR T cell, if you use some approach to get rid of senescent cells, that ought to be good for an old organism. And that seems to be true in animal models. Whether or not that's going to work in humans, I think is still an open research question. Next slide, please. So let me briefly cover these three set points. Next slide. So we looked at this a long time ago by just sort of downloading all the GWAS studies that were linked to phenotypes that were age associated. And we were actually all human phenotypes that were not sort of congenital things like, red hair or something like that, but, things that were associated with human disease to see if there was a locus in the genome that was hit recurrently over and over again by genome-wide association studies as being associated with multiple diseases. The logic being that if aging is associated with multiple diseases. So if there were a locus of the genome that were important in aging, it should be associated with multiple diseases too. And the answer to this is a very clear yes. Next slide. And you can see that analysis sort of, you get really two main loci. 6p21, which is the MHC locus, there are 4 contiguous bins there. Each one of these bins is 200 kb. That are associated with a variety of human diseases, and I mean hundreds of human diseases, and they're all things like rheumatoid arthritis and scleroderma and autoimmune disease and type 1 diabetes, for example. And then the other major hit you get is the 9p21 locus, which is the INCARF locus, or also called the CDKN2A locus. It's known to cancer researchers as the melanoma, glioma, pancreatic cancer, germline susceptibility locus. But it's also through GWAS been linked to just about every age-associated disease one can think of, including atherosclerosis, stroke, glaucoma, cancer, type 2 diabetes, etc. The only real strong age-associated disease is not linked with is, to my knowledge, Alzheimer's. But excluding neurodegenerative disorders, it's strongly linked to many age-associated diseases. And I think the idea here is that the beneficial, anti-cancer function of this locus that promotes senescence, it includes P16, the perhaps gene best associated with the induction of senescence in vivo. The good thing is P16 induction prevents these cells from becoming malignant, and people who don't do that very well are associated with germline cancers or germline cancer susceptibility. But the bad news is that the activation of this locus has these untoward effects for other diseases like type 2 diabetes and atherosclerosis. Next slide, please. And that's just showing these things at various linkages. Again, all sort of age-associated conditions. Next slide, please. So, by the way, that analysis has now been held up with more and more GWASs, and it has been done by lots of other groups, and a molecular understanding of how those specific SNPs are associated with activation of the locus is now reasonably well understood, and it's an exciting topic, but I'll leave it there for those of you who want to follow up further on your own. Let me now talk about measuring aging, especially in cancer-treated patients. So next slide, please. So the idea here was that you wanted to somehow measure senescent cells, and we spent a lot of time trying to figure this out and developed a T-cell based assay because we really wanted to have a blood test one could do. T cells express P16. As I said, P16 is the sort of strongest marker of of senescence that we have in vivo. It's been used for a variety of purposes in animal models related to aging research. And so we developed this test where we purify your T cells and then measure P16 by RT-PCR. And that sounds really complicated and not like the basis for an analytic test. And that's largely true. This is a very difficult thing to do for clinical use. It's relatively straightforward to do for research use, but standardize an RT-PCR test of a blood-based specific cell type is complicated for a lot of reasons. Next slide. But it does work, so we developed this assay. We did it in thousands of people. Eventually a company tried to commercialize this test, so it's been done. We're still working on it. It's been done now in many thousand people. And basically it correlates with some aspects of T cell aging, and it is associated with certain clinical syndromes. And that's really, the idea is, can this be a biomarker for physiologic reserve? Could you measure someone's P16 prior to, say, surgery and tell if they were going to recover well, or prior to chemotherapy and see if they're going to bounce back? So that's really the idea here. I think a test to measure senescent cells in vivo would be useful for that purpose. Although, to date, the ideal test for that is still met with a lot of technical challenges. Next slide, please. But here's sort of what you get when you do this. You see, in a research use that the increase in P16 with aging is exponential. So, it's a log scale. So for every, it's sort of it's a 20-fold increase over 8 decades. There's evidence of aging well before people are aged. You can look at people who are sort of molecularly young at any given chronologic age versus molecularly old and do phenotypic observations. This is a much stronger correlation with aging than other biomarkers such as telomere length, although a less strong correlation with aging than biomarkers such as DNA methylation. But perhaps useful for measuring the burden of senescent cells. That's what we've hoped it estimates. Next slide. This has been tested in a number of clinical scenarios. I won't talk about the non-malignant uses, prior to, kidney insult or these kinds of tests, but I'll focus briefly on some of the cancer ideas. And that gets back to this idea that chemotherapy promotes aging. So we know that from animal model research. There's been a variety of literature through the years that have established that, age-induced phenotypes occur in response to DNA damaging agents. Next slide, please. So we tried this in people, and the idea was to measure people's blood levels of P16 prior to adjuvant chemotherapy for breast cancer. And this is patients that were treated kind of with the physician's choice of breast cancer, but the regimen had to be at least 4 cycles of an anthracycline and cyclophosphamide, which is a fairly heavy-duty DNA damaging regimen. Next slide. And then we measured molecular age afterward at 3 different time points, and it turns out it doesn't matter which time point you use, as long as the counts have recovered. Any time point after will do. And so we just grouped them all together for analysis. Next slide. And this showed, again, plotted on a log scale, that just about everybody who gets cytotoxic DNA damaging exposure will have an increase in their expression of P16 in peripheral blood T cells. And we believe this is a surrogate for the whole body burden of senescent cells. And you can see, on a log scale is a fairly substantial increase. Some people's P16 goes up remarkably, others people stays sort of flattish, but just about everybody had an increase. There was one individual who did not. Next slide, please. Yeah, and so 4 cycles of AC is sort of on the order of 15 years of chronologic age, if you line that up. And I don't think this is surprising to oncologists. We know that people who've gotten a lot of DNA damaging agents in the past have a markedly increased later lifetime risk of myelodysplasia and leukemia and the diseases of aging of the bone marrow that are common. So, the fact that a person who's gotten breast cancer therapy has their bone marrow remains old in a way after they recover is, I think, well known to clinical oncologists. Next slide. So let me now talk lastly about this idea of senolysis. This is, I think, originally was pioneered with small molecules to get rid of senescent cells, but now as a number of commercial approaches are being adapted. I can say the real champion of this has been Jim Kirkland at Mayo and Jan van Duurzen at Mayo, who did both human and mouse experiments to sort of test this idea out. Next slide, please. And the idea is that if these cells that are senescent, that persist for decades, if not forever, are somehow cleared, then they won't make those cytokines they used to make that are bad. They won't, obstruct anatomic niches the way they used to. And And there are a number of approaches. So in mice, the number one approach is to use the P16 promoter, which, as I said, is a marker of senescence with aging, to drive toxic genes like the attack gene or this 3-MR gene, which allows you to give a drug to the animal, which will then kill senescent cells. But then there are also small molecule approaches that are being tested in humans. And there are a number of clinical trials now going on with a variety of small molecules from, mostly biotech companies that are interested in anti-aging therapy. There are immunologic approaches. And I'll say, we worked with a few other labs working on this topic, and the benefits of senolysis vary significantly by experimental system. So, the genetic clearance would give a different result from in a tissue versus using a small molecule approach in an animal model. Next slide. One quick result we did, we actually made a mouse that overexpressed a reporter gene, the tomato reporter, in P16 expressing cells. So you could look in vivo to see if there were senescent cells and if you were able to clear them. This is harder to do than it sounds, and this reagent is not perfectly well suited for this. Next slide. But it does work in some compartments. And so here's an in vitro result using chondrocytes. It turns out that cartilage makes a lot of P16 with aging, and that's an interesting finding. And they certainly in culture do as well. And so you can see on the x-axis here is tomato expression, and in early culture, cells don't make the reporter, the tomato expression, but with culture, they become senescent and start to do that. You can see a big expansion in the senescent fraction to about 1/3 of the cells in culture. And then treatment specifically with the senolytic, next slide, will deplete those cells. So there is, and navitoclax is a BCL2 inhibitor that was initially developed for cancer usage, but is one of, if not the best studied senolytic, it's close to it. And has been as one of the compounds being tested in humans. Next slide. And here's the sort of result that Dr. Van Duersen reported in mice. And you can see these animals are litter mates. They're the same age. And the mouse on the left is old and frail because it had natural aging. And the one on the right is the same age, but was treated with senolytics to reduce the number of senescent cells. And it's much healthier and happier. Next slide. And it lives longer. So mean lifespan and median lifespan of the animals are increased by senolytic treatment. And so this is really, as I said, led to a bunch of commercial efforts now. But I think it's too soon to say whether or not the paradigm of senolysis will pay off in people. Next slide. Just remind you, senescence, this can work after chemotherapy. So an idea of Judy Campisi was to try and deplete these senescent cells that are induced by drugs like anthracyclines and see if that would help patients. And it really sort of survivorship research, if you will, that patients who have long-term toxicities for chemotherapy might benefit from a senolytic approach, which I think is an interesting idea. And in many ways, testing these kinds of approaches in patients who've recovered from cancer has a number of clinical trials advantages, if you think about it. Next slide. So let me talk now briefly about hematopoietic stem cells. Next slide, please. So hematopoietic stem cells get old as well, but we think for a different reason. They don't undergo senescence. They actually have a change in their epigenetic state whereby they lose their ability to make progeny. And so damaged or old hematopoietic stem cells, if you will, prefer to self-renew, and they can still divide. They're not senescent, but they don't make blood cells very well. And that is, as I said, reproducibly caused in both mice and humans by serial treatment with DNA damaging agents like chemotherapy. And this is an old finding is of widespread clinical importance. Next slide. And we decided we wanted to work on this, and this requires a trick of the cell cycle. And so when I started out in my lab, this is how we thought the cell cycle worked. And this model is totally wrong. Every prediction this model makes is wrong. Next slide. And now we have a much more nuanced understanding of the cell cycle and understand that there are a variety of different kinases, CDK4, CDK2, and CDK6, that will drive proliferation. And importantly, what that means in practical terms is that drugs that inhibit CDK4 and CDK6 are potentially useful for human treatments. In the old model, when you thought every dividing cell needed CDK4 and CDK6, that would be a miserably toxic drug that would stop all cells from dividing. But that turns out to not be the case. I think anyone who treats patients with breast cancer is very familiar with palbociclib, which is well tolerated by patients for years and years and years and doesn't cause certainly widespread block and proliferation because humans have other kinases they can use to drive the cell cycle. So that was a basic science discovery, largely attributable to Peter Szynski and others. that I think really had important implications for drug discovery. Next slide. So here, when we heard this was possible, we got palbociclib, which at that time had a number. It was like 991 or something from Pfizer, and just tried to learn what cells in mice were sensitive to CDK4 and CDK4/6 inhibition. So what cells needed these kinases to divide in an adult animal, and what cells didn't. And so using this brown marker, which is a marker of proliferation, you can show that beta cells in the pancreatic islet need CDK4/6, and some of the cells in the pituitary need CDK4/6. And lymphocytes, both T cells and B cells, it turns out, need CDK4/6. But that's kind of it. There aren't a whole lot of cells in an adult mammal that need CDK4/6 for proliferation. Most of the proliferating cells in the bone marrow and in the small bowel and the gut divide just fine. And you could put these animals on these therapies for months, and they would have no terrible gut toxicity, and they'd have only modest myelosuppression from this. So it was well tolerated, and I think certainly speaks to the reason why CDK4/6 inhibitors have been successfully used in people for so long. Next slide. So this meant to us that you could modulate the proliferation of some cells with minimal toxicity, which is an important finding, and I think that's the basis for why palbociclib has been successful in ER-positive breast cancer. That ER-positive breast cancer cells often need CDK4/6 for proliferation, and blocking it is useful. But it also meant something else that we wanted to try in my lab, which is that you could protect cells from DNA-damaging insults by inducing a cell cycle pause. So, and particularly the cells we had in mind were hematopoietic progenitors, that you can make these cells not divide. That would be good, that those cells would then be resistant to DNA-damaging agents, which would be a benefit. Next slide. So I've sort of listed on the left here things where you might want a CDK4/6 inhibitor to block the proliferation of unwanted cells. And, CDK4/6 inhibitors are well established now in cancer, are being countenanced for use in other clinical syndromes like autoimmune disease, for example. And on the right are cells you might want to protect from DNA-damaging insult. And the hematopoietic progenitors and renal epithelia are two that come to mind. And I won't talk about the kidney work at all, but suffice it to say, kidney cells generally don't divide until they're damaged, and then they divide a lot, and they require CDK4/6 for this for this activity. And so they are potentially cells that could be protected from nephrotoxic agents as well. Next slide. So you need a new drug for this. Palbociclib would not be the ideal approach for this, because for a variety of reasons, not the least of which is one would want a drug with an IV formulation. We spent a lot of time trying to develop, trying to license a CDK4 inhibitor for this, and eventually ended up making our own. Next slide. And so that's a story of a molecule called trilaciclib, which used to be called G1T28 and is now called Cosela, with its recent FDA approval. And it's very, type 1 kinase inhibitor, looks a lot like ATP, blocks CDK4/6 activity with, picomolar activity, is very, very effective at inhibiting the proliferation of RB-dependent CDK4/6 or RB-competent CDK4/6 cells, but not RB null cells. So tumors that don't have RB will divide in the presence of this molecule without any problem. Next slide. And IV formulation, so it's the kind of drug you could give prior to the infusion of chemotherapy, put the cells in G1 or G0 so that they won't be damaged. And then as soon as you stop the infusion, the drug goes away and the blood cells begin to proliferate again. It's exactly what you would want for a drug used for this purpose. Palbociclib has a multiple day half-life and would be less favorable for this. And it's an oral drug. And if someone throws up their pill before chemotherapy, that would be problematic. Next slide. So here's a list of the CDK4/6 inhibitors that I'm aware of. There may be more than this now. Trilaciclib is shown here. There's also palbociclib, ribociclib, abemaciclib, and then lirociclib, which has now been out licensed to EQRx. Trilaciclib is the only IV one. You can see they all are very, very potent for CDK4 and 6 versus CDK2. That turns out to be really important for their use. And abemaciclib is a little different. It's not as selective as the other four, and has a quite different structure from the other four. It is also the only one that appears to have good CNS penetration, which is, I think, part of the basis for its success in breast cancer. Next slide, please. To briefly show you how this works with hematopoiesis, you have to remember how blood cells come about. Basically, the hematopoietic stem cell is quiescent. It seldom to never divides. It very infrequently proliferates unless the blood cells are damaged, in which case it is coaxed through the cell cycle. Most of the work on a daily basis is done by these progenitor cells that give rise to myeloid cells and progenitor cells, and also lymphoid progenitors, and they do that all the time. And this is not shown to scale. There are sort of like 20 proliferative events between these cells and the differentiated in cells. But Most of this proliferation shown here, as I already mentioned, does not require CDK4/6. It's really only the sort of the progenitors and the lymphocytes that use CDK4/6. The rest of the cells will divide just fine in the presence of a CDK4 inhibitor, which is why people taking palbociclib don't get, terrible myelosuppression. Next slide, please. So this is where we showed that explicitly. We looked at the ability of trilaciclib to inhibit the proliferation of various hematopoietic compartments and show that these LSK cells, these hematopoietic progenitors, which are, very pluripotent cells, are really, really sensitive, as are T lymphocytes, and most everything else is not. You can see on the right how long this lasts. So if you give mice a dose of trilaciclib, the cells go out of cycle at sort of starting around 6 hours and remain out of cycle for about a day, and then they start dividing again. Next slide, please. It's been known since the 1960s that cells that are in G1 or G0 are resistant to DNA damaging agents. That's shown here that basically if you expose cells to radiation right around the G1 to S transition, they are sensitive, but very, very resistant at the beginning of G1. And if you can lengthen the period of G1, you can extend their period of being resistant. So the idea was to take these proliferating cells that are hematopoietic progenitors, put them all in G1, and then give the DNA damaging agent, be it radiation or chemotherapy, and then thereby spare the cell. Next slide. It works pretty well in mice. So here is an experiment in animals where you show that all lineages, red cells, neutrophils, lymphocytes, the gray is untreated mice, the blue is animals that got 5-FU alone, and then the red is animals that got 5-FU after trilaciclib. And you can see all cases trilaciclib preserves the blood counts. Next slide. Hematologic toxicity is a big deal. I think it's very familiar to oncologists. It causes significant morbidity in patients. It's very expensive. It causes febrile neutropenia with emission and the use of growth factors, which are expensive. And it delays therapy. It leads to reduced dose intensity, which compromises the efficacy of chemotherapy for diseases like triple-negative breast cancer and small cell lung cancer. So this is one study from Mark Szynski using etoposide platinum in small cell, and you can see, the vast majority of patients with that regimen have significant hematologic toxicity. So doing something about it ought to be beneficial. The other reason we were particularly interested in small cell lung cancer is small cell lung cancer is always RB null, and therefore one can treat humans with small cell lung cancer and protect their bone marrow without any concern that you'll protect the tumor. The tumor will cycle merrily on. Next slide. So here's how all the trials were done. You can see the NCTN numbers up there. These are the ones in small cell and triple-negative breast cancer. They generally involve chemotherapy with or without trilaciclib in a randomized phase 2 design. The endpoints are typically hematologic toxicity, but secondary endpoints are always, outcomes. All of the trials have been positive. There's a survival advantage for triple-negative breast cancer and significant myeloprotection for small cell, either in the first-line setting or the second-line setting. Next slide, please. Here's one of those trials. This is the first line, etoposide platinum in small cell, and you can see that a big reduction in neutropenia and dose delays and GCSF usage. And so all the lineages are really protected by this approach. It works well. The estimate now, the number needed to treat for a blood transfusion is like 3. Like 1 in 3 patients will be spared a transfusion if treated with trilaciclib at the time of therapy. These data in aggregate were submitted to the FDA. The drug received FDA breakthrough designation and priority review. Next slide. And was approved with brand name Cosela last week, or 2 weeks ago. For so far, extensive stage small cell lung cancer, but I think the paradigm would be useful for other RB deficient malignancies as well, like triple-negative breast cancer, for example, or HPV-associated head and neck. And so now I think it's really interesting to try and look at this ability to intensify therapy in a variety of cancers would be a benefit. It's a way to do dose intensification without doing stem cell transplant, for example. To be clear, by the way, when I entered federal government, I completely divorced myself from the ongoing operations of this entity. So everything I know about the operation going on today is public knowledge. And I've, of course, fully divested of all, entanglements. Next slide, please. And, to get back to aging, the whole reason we started doing this is because I've always thought of this as an anti-aging approach. Really, the idea here is to protect hematopoietic stem cells from the toxicity of chemotherapy, to block that iatrogenic aging, if you will, and then thereby patients would benefit in the long term. So there'd be reduction in the long-term toxicity of DNA damaging agents. And as I said, that's pretty easy to establish in people, the acute toxicity, relatively easy to establish the decrease of acute toxicity. But the long-term analysis is a much harder thing to do. And to date, really all the benefit has only been shown in animal models. So here's an animal experiment where we serially treated animals with 5-FU to exhaust their bone marrow with or without trilaciclib. And then we looked at the ability of the hematopoietic stem cells to do stuff like make blood and make lymphocytes, which turns out to be a defect of old HSC. And we showed that the co-administration of this drug would not only reduce the acute toxicity of 5-FU, but it would also reduce the long-term toxicity on hematopoietic stem cells. Next slide. Here's a little bit of that data. You can see that the untreated, these are competition experiments where one transplants HSC that have and have not been treated. And you can see in untreated mice, sort of 50% of the bone marrow is made from the control cells. But when you give animals 5-FU, their ability to compete for the donor percentage declines, and that is rescued, not completely, but substantially by using trilaciclib. It is not rescued using Neulasta, G-CSF. In fact, Neulasta makes hematopoietic stem cell function worse over the long term. So this is the hope now that if we could prevent the acute toxicity of chemotherapy, one also could subsequently reduce long-term things like myelodysplasia and leukemia, secondary leukemia. Next slide, please. So let me summarize this part to say that cytotoxic chemotherapy cures cancer, and as an oncologist, I deeply believe in the administration of adjuvant chemotherapy and people with advanced disease, and I think it's very, very important. But it also has some toxicities. It causes senescent cells to accumulate, which drives some of the long-term toxicities. It causes stem cell exhaustion, which has important clinical consequences. And there is now at least one approach which I think has some promise to ameliorate some of the long-term stem cell toxicity of cytotoxic, of this sort of iatrogenic aging, if you will. Next slide, please. I thought briefly before I wound up, I would talk about where the NIH is in all this. So the National Institute of Health is very interested in aging research now. The NIA's budget, the National Institute of Aging, has substantially increased both for Alzheimer's research and non-Alzheimer's research in the last few years. And the NCI works closely with the NIA. I used to be on NIA Council. So for many years, I was an advisor to the NIA. And I still, Richard Hodis, the director of the NIA, is someone that the NCI director, I speak to regularly. And so we have sort of some partnership grants now that are intramural, extramural with the Waxman Foundation, for example. We have a cancer and aging interest group, next slide, that has, convened some meetings. We have a notice of special interest, which is sort of, it's a PA, if you will. It's a program announcement, and it's the new lingo, to ask for grants studying the relationship of cancer and aging, of survivorship issues, statistical modeling, interventional trials to reverse or mitigate age-associated effects, particularly in patients with cancer. And that NOCI is open until March 8th. Next slide. There is an important human tumor atlas program going on as part of the Moonshot, which is to try and really identify rare cell populations in all organs in old and young people. And so you can imagine senescent cells might be the kind of thing that we would find in the human tumor atlas to accumulate with aging. And that's one of the hopes of HTAN, is that we'll start to understand at the sort of cell by cell level what's going on with aging in various organs. But we felt that this effort, which was pioneered by the NCI, wasn't really sufficiently focused on senescence. And so now there's, next slide, a new Common Fund initiative. So the Common Fund is, NIH funds. This is Francis Collins's money to support a new initiative on understanding senescence across organs, across disease types, not necessarily focused on cancer, but focused on other, senescence-associated disease conditions. And those RFAs are now on the streets. There's a network for U54s and a coordinating center, and it's $200 million program over several years. So for those of you who are interested in aging and cellular senescence, importantly, I would recommend you look at these RFAs. They, I think, are a tremendous opportunity to get involved in network-based research with Common Fund support. Next slide, please. And lastly, one of my long-term collaborators in this space, in clinical research, I deeply relied on people like Hymus, but also for many years, Artie Huria, who I think was really one of the oncologists interested in geriatric medicine, who understood the implications of things like stem cell aging and senescence and why they might be important in patients, and how could one use clinical trials. I'm sure this audience is familiar that Artie recently passed away in a tragic accident. But we have a seminar series in her name, a memorial lecture series going on at NCI. Here are a few of the speakers. It's been a really exciting group of talks, and I think really honors Artie's legacy in an important way, given her a strong commitment to older patients with cancer for many years. Next slide, please. And with that, I'll close. I hope I have time left for questions. I didn't mean to go long. I think I tried to end on time, if there are any. So thank you again for having me. And this is my favorite topic to talk about. So look forward to working with the group.
Ned, thank you, Dr. Sharpless, very much. And let's open this up to questions. And there are two ways to ask questions. You can either put your name or question in the chat room, or you can raise your hand. So while people are entering all their names and questions, I'll throw a question out to Dr. Sharpless. So Ned, you talk about the impact of chemotherapy and how it impacts cellular senescence, but there's other ways we kill cancer cells as well. And so whether we're talking about a kinase inhibitor in lung cancer, or we're talking about a PD-1 inhibitor in melanoma or renal cell, those also kill cells. Do they have similar impacts on cell senescence, and would they be, and have you witnessed or observed that the aging phenomena, which I think as a clinician, I absolutely say is there with chemotherapy, I think it's probably there a little less so with immunotherapy and with targeted therapies. Just curious what the molecular mechanisms might suggest, and do you see this in other cancer therapies?
Yeah, so we're very interested in both directions. Does aging affect the ability of the therapy to work? And does the therapy make you old? Right. So we've looked at this both ways. In animal models, this has been done a lot. And most of the agents that you think of as being non-DNA damaging, or even some of the DNA, like taxanes, don't seem to be as bad as anthracyclines, for example. So agents very much differ in their ability to promote senescence. There may be other ways cells can grow old that are important too, but with regard to cellular senescence, drugs like Cytoxan, etoposide, platinum are really bad. Taxane's less bad, and then kinase inhibitors and checkpoint inhibitors don't seem to do much at all. I think the opposite direction is an interesting question too. I would have predicted when I started that checkpoint inhibitors ought not work as well in older patients, because the T cell organ ages significantly. I mean, it really changes behavior. The milieu of T cells changes, the sort of repertoire of diversity of receptors changes markedly, their proliferative ability changes markedly. So I would have thought at some stage checkpoint inhibitors would stop working, but that may happen, but it's very minimal. I mean, there are well-documented cases of, Jimmy Carter, 90-year-old individuals responding beautifully to checkpoint inhibitors with robust T cell immunity. So it's hard to predict. I don't think we understand the intersection of aging and checkpoint inhibitors yet very well. The one area where I do think this is going to be big, frankly, is in cellular immunotherapy. I mean, there's a reason why those trials were immediately positive for kids with ALL and not so good for CLL, and for 70-year-olds with CLL, and it largely has to do with the ex vivo proliferation of lymphocytes, and that drops off steadily in an unpredictable way. And so I think if you can really understand what about a T cell makes it old, or what fraction of T cells you need to culture ex vivo, cellular immunotherapy will improve, because the manufacturing challenges are still quite significant. And it ought not be that difficult to pick non-age cells or to do something to the cells to make them less old, epigenetically, to allow ex vivo expansion better. But still, if you look at patients that have been heavily pretreated with chemotherapy and you try and make T cells, often it fails. And we don't know why. I mean, that seems to me a perfect model to study the cellular basis of aging, because it has important clinical consequences for the patients, but it's still a little bit of a mystery.
And along those same lines, how about radiation?
Yeah, well, so all the cytotoxic stuff I talked about in terms of acute toxicity, hematopoietic progenitors is true with radiation. In fact, that's how we got into business. We did all the experiments we did radiation first because it's a lot easier. There's no pharmacology for radiation. You sort of, when the beam is on, that's when the damaging exposure occurs. So it's a simpler system than trying to do this with etoposide or 5-FU or something, which took a lot of work to work out those animal models. So radiation is very much depends on cell cycle to kill. And so we think there's a real opportunity to prevent radiation-induced toxicity to myeloid progenitors using pharmacological quiescence. And also, by the way, I think this is a kind of a DARPA, or a DoD thing, but one can imagine that you could also use this approach to reduce the toxicity of inadvertent radiation exposure. Say if there were a nuclear disaster, or that by putting stem cells into cycle at the time of an inadvertent exposure seems like it ought to be a good idea. And I've learned a lot about how radiomitigants and radioprotectants work in terms of their clinical development, and it is not straightforward, and it would be a topic for another day. But suffice it to say, I think this would be a promising approach. It works really well for chemotherapy, and chemotherapy is a lot harder to do than radiation. For aging, the link between radiation and senescence is very strong, and that is the first experiment's really done. It's been done by multiple groups. You can show interesting things like, if you radiate someone with head and neck cancer, you can find senescent cells at the port, but not at the nearby skin. So there's very good evidence that DNA damage will directly promote senescence with lifelong changes in that organ. And as all of us are familiar with people getting some late radiation toxicity 40 years after their radiation. Everybody's got stories like that. So I think radiation does age tissues in a way that is memorable for the cell at a cellular level. We think that has something to do with senescence, but not entirely. By the way, I think Ross Levine is coming to talk, and he'll talk about a different model of aging that involves genetic mutations of hematopoietic compartments, CHIP, clonal hematopoiesis. And I think that's really cool and important, and probably at work in tissues beyond hematopoiesis as well. And that may be a different way. So there's sort of epigenetic aging, there's senescence, and now there's genetic aging. There are three different cellular bases for aging, and certainly I believe all of them are overlapping.
Nancy Davidson, question?
Good morning, Ned. Ned, is there any natural product out there that you think is a bona fide senolytic? Our patients wonder about this all the time.
Yeah, so I was very skeptical of the senolysis thing. I thought it looked too good to be true. As an oncologist, we all have experience of things that are touted in the media and then don't work in our patients. So and then my lab was actually approached by Daohong Zhou, who had been a collaborator for other reasons, about helping him measure senescence using P16 and in vivo models to test out. Daohong had identified navitoclax from a screen. He'd screened 100 compounds of known mechanisms of action, and navitoclax was really his only hit. And a really strong hit, though. I mean, it kind of makes sense because senescent cells are damaged, but they don't die. They suppress apoptosis in some specific way. And so the idea of using an activator of apoptosis, it made sense in retrospect, but that certainly wasn't why Daohong did the experiment. So we had these phenomenal in vitro results, and I was very skeptical, but we did the experiments blinded in my lab and saw a very, very compelling senolytic effect, which we published in a Nature Medicine paper that's, was the basis for those human clinical trials. It's very compartment specific. It doesn't kill all senescent cells. There's pharmacology issues. There's sensitivity of the different, there's not even a great pharmacodynamic marker in people yet, making these trials very, very challenging. Now there are a number of other small molecules that are being developed. There's a company called Unity Biotechnology that has p53 activators. And I think there's a company that's developed, immunologic approaches that has developed, cellular approaches to kill off senescent cells. How these are going to work, I don't know. The available sort of supplement, if you will, is dasatinib, which is a drug, but also in concert with quercetin, which is something you can buy at GNC as a dietary supplement, if you will. It is not a controlled substance. And that comes from Jim Kirkland's lab. He tells me it doesn't work very well if you just take quercetin alone. You have to combine it with dasatinib to really get the effect. So most patients won't have that available to them. I would not recommend prescribing something like that off label. I think that is needs to be tested in a formal clinical trial. But Jim, to his credit, a real geriatric oncologist has trials going on, or geriatrician rather, has trials going on. So he is trying to promote renal reduction in long-term renal toxicity, for example, using that combination and other measures. So we'll know. I mean, that's what I always like to say about the senolytic hypothesis is it's very testable, right? I mean, there are molecules that do it, and there are clinical trials approaches, and if it works, we'll know pretty soon. And if it doesn't, it'll, like the other 150 theories of aging that didn't pan out in patients, it'll just go away. Yeah. But I really don't like theories of aging that are untestable, like the reactive oxygen species theory of aging, which has got no direct experimental support for that, but it's still, why we drink pomegranate juice and stuff. You know, it's impossible to make that theory go away, despite the fact that there's no evidence it's actually true. So senescence will know. If it works, it will know. And if it doesn't work, we'll know why.
Ned, thank you. Next is Dan Gotchling. Dan?
Hi, Ned. I was wondering if the SAS aspect of senescence impacts the hematopoietic stem cell niche. Because if it does, then that might have a positive feedback loop that's harmful, right? So do you have any information or knowledge about that?
There are people working on this. It was not a focus of my lab, but you know, early on we thought hematopoietic stem cells themselves underwent senescence. There were claims to that effect by a few groups. Those largely went away with more careful studies. That, you know, those experiments largely date to the period before hematopoietic stem cells could be isolated to single cell purity. So when you're looking at populations of cells, you would find evidence for senescence. But when you really got to, you know, SLAM CD150 type purification, it became clear that HSC don't undergo senescence with aging. And so, you know, people like Sean Morrison and Derek Rossi kind of killed off that idea. But what was interesting to me about that is if you look, there is a cell pretty close to HSC, and we think it's the lymphoid progenitors, the very early, you know, common lymphoid progenitor, the CLP. That cell probably does undergo activation of P16 and something like senescence. It kind of drops out with aging. You don't make those cells anymore. And we think that's because the senescence machinery, when turned on, will deplete those cells. And that's largely work from Ken Dorshkin, a real professional lymphocyte biologist. So now, but you know, HSC don't work very well when you get old. And so why? And one of the questions has been, is it a niche thing? Is it the milieu about those cells? Or other, you know, there are lots of cytokines that are at different levels with aging. So is it, you know, the both either the paracrine or endocrine circulation of such hormones that has an effect? And there have been lots of claims both ways, as I'm sure you know. There have been parabiosis experiments that claim a marked reversal of HSC aging. Those experiments have been done in multiple labs with different results. So I don't know what to believe. I mean, I think that I will be very, very surprised if there isn't some endocrine effect on HSC function with aging, given that there's, you know, everything is different in an old versus young animal. But which one is important and whether that's the hematopoietic niche, I don't think it's still known.
Thanks.
Next is a question from, and I'm sorry for not recognizing the name from the call letters, R. Prentick. It might be Prentice. Is our Prentice still? Well, I'll read the question of our, there she is.
It's Prentice.
Ross, you're on mute. Ross, you're on mute.
Here we go. Sorry about that. So Tom, I asked the same question you did at the end about radiation therapy and implications for the slowing of aging through the devices that Ned was talking about. So it's been answered. Thank you.
Okay.
But maybe I'll say a little more about that though, because I don't think I really answered it fully. So, you know, if you can't, so we're talking about the ability to reduce the long-term toxicity of, you know, like we do with myelosuppression. If you can't make the cell go out of cycle, you can't protect it, right? And so it turns out that lots of the dividing cells are CDK4/6 insensitive, like cells of the gut. So we never really were able to get good gut protection from radiation because the cells just cycle and they're still sensitive to radiation. The hematopoietic progenitors are relatively unusual. There have been other groups that have claimed differently. They claim that there are, you know, brain cells, you know, like hippocampal sparing kind of thing, or there are, you know, squamous cells of the head and neck might be CDK4/6 dependent. And I already mentioned one that we know is CDK4/6 dependent, which is the renal epithelia. So I think there may be an opportunity like to reduce platinum-induced nephropathy. So, you know, cisplatin has a very high frequency of damaging the kidneys, and we think that that is something that one could reduce with a drug like trilaciclib. That was based on experiments with Ben Humphreys, who's now chair of renal at WashU back in the day, and it works pretty well in mice. But you know, that's a relatively uncommon toxicity now. It's hard to test that in humans, and I don't think it's been a high priority for the developers of trilaciclib, although maybe you'd find that in a large enough study of small cell, for example. But radiation, you know, the one thing we know would work is you could reduce radiation-induced myelosuppression. That will work. We've done that in mice, you know, when given TBI, total body irradiation. most human radiotherapy fields are not large enough to cause myelosuppression unless they're intended to kill off the bone marrow, like a conditioning regimen for transplant or something, in which case you wouldn't want to protect the cells. But, there are still, I'm told, like sort of half body radiation regimens and, full upper torso and stuff like that, where myelosuppression is a deal. They're rare, but, you could imagine trying an approach like this in that kind of a treatment regimen. I think is an interesting idea. Now that the drug's approved, I hope people will take up clever usages like that. It's easier to do clinical trials.
I was thinking particularly of prostate cancer, which maybe the field is intermediate size.
Yeah, the other problem you got to keep in mind is if you got to treat them every day, CDK4/6 inhibitor IV every day for 40 fractions would have toxicity of its own. So you would think this would be better with these very high dose, flash type regimens of high dose radiotherapy with short treatment fractions. I think it's an interesting idea. I hope somebody does it. I don't think it's a commercial opportunity for a biotech company, but I think it's a very important clinical question.
I think your point, Ned, about the frequency of treatment with radiation, the difficulty in small cell. I have a relative who's had 5 red cell transfusions and 2 treatment delays with limited stage small cell, and going through treatment right now. And having seen this last week, I thought, gosh, this would have been a great option for her. But then thinking about a daily infusion would be a bit of an issue. Well,
no, you only have to give it with the time of the drug. So it's the 3 days of etoposide.
So what about the radiation?
Oh no, see, radiation, yeah.
Because she's getting radiation, it's limited stage. So
this is a patient who's getting concurrent or
concurrent chemorads for limited stage. So
that's why it's approved for extensive stage.
No, I know. That's why you're stuck. All right.
Yeah, I don't know how you would do that. I think there might be a way to do it, but somebody's got to do the trial and figure it out. But it's not obvious how you would give the drug.
Well, you got Phil Greenberg's attention when you mentioned T cells. Phil?
Yeah. Oh, there I am. Hi, Ned. Good to see you. So I'm curious, the T cell assay you're talking about, which was giving you some insight into the relative aging or senescence of an individual that you're using as a reflection. Is that merely a reflection of the changes in, although I'm not clear what's chicken or egg here, but that it's merely a reflection of the T cell subpopulations that you get out of the person. So if you actually isolated central memory cells or memory stem cells, would they look essentially much more normal than if you do the whole population as you were doing?
Yeah, Phil, good to see you again. I don't exactly know the answer to this question. I hope that as more and more people do, single cell sequencing of the T cell organ with aging and start publishing those data, it will become clear. I can tell you what we did. We looked at CD4 versus 8, and both of those expressed P16, 8 more so than 4. We looked at these markers of exhaustion, and they did not correlate with markers of senescence. We think that exhaustion and senescence are just distinct things. I don't believe today, right now, you could unequivocally say, I have in my hand a senescent T cell. I think you could find T cells that make P16. That's not hard to do, but they divide less. That's true, but they don't have a lot of the other characteristics of senescent cells. They don't make lots of, they make a little bit of IL-8 and like nothing else. So they're not really like the senescent fibroblasts that are beautiful and have every phenotype you can imagine. So, whether or not that's real in vivo senescence, we don't know. What we think is going on in that experiment, it's not that, there's a 10-fold increase in P16, but we think what's really happening is 1% of the cells have 100-fold increase or, 1,000-fold increase. So we think it's very dramatic activation in a few cells that are T cells that are CD3 positive. But the majority of the cells are not expressing P16. So what does that mean? Are those cells just an epiphenomenal marker that sort of correlate with aging and they're useful as a kind of clock, if nothing else? Or do they have toxicity because they make hormones and they occupy niches? We don't know. I do think that the antibody-based detection of P16 with aging has been very challenging. So I think it will require single cell RNA, but I think those experiments, like in HTAN, I mentioned the human tumor atlas, will get at to the bottom of this. But I will say, though, that I don't think it's completely understood to my satisfaction why it is harder to make some of these cellular products from older people, whether it's a change in some population or the per cell ability is decreased. And I don't think that the people who just say, oh, that's exhaustion, we understand that so well, really know what they're talking about, because they describe a phenotype that's basically cell doesn't divide, but they don't know why, and they can make them divide sometimes, but not always, and sometimes they make p16, but not usually. And so I will admit we don't understand it very well, but I don't think anybody else does either.
And then a question that comes up is the and this may just be my naivete about it. The differences between longevity and aging at a molecular level. So, calorie restriction, metformin, my impression is have been among the only things that have been shown to prolong longevity. How does longevity molecularly compare to the principle of aging? Obviously they're related, but what is do we have any sense of whether mechanistically there's anything there?
It's a really complicated topic. I think there are animal experiments that give fairly clear answers, but their human correlates are often confusing. I'd say that the strongest small molecule data in rodents is with rapamycin. I mean, that's very good at extending lifespan. It's been reproduced by multiple groups. It works in multiple species. And rapamycin is probably too toxic to give people for 30 years or whatever to try and do this. So there's now a hunt for rapalogs that have less toxicity, but may still have the anti-aging effects. But the problem is nobody knows how rapamycin works. A surprising result, by the way, that has now been validated by many groups, I think it came out of Judy Campese's lab initially, is that rapamycin attenuates the SASP. All those senescent hormones are greatly reduced with rapamycin treatment, which may have something to do with its immunologic functions. So, it could be an anti-senescence drug. People always said, well, gee, if there were a drug that would attenuate the SASP, that would be great. And lo and behold, we had one the whole time. But it's also, rapamycin has a range of metabolic effects that are very, very important, which might be really how it works. Metformin, as far as I know, has no effect on senescence. There are other small molecules that are like aspirin-like molecules that will extend rodent lifespan. We tried aspirin in people under some of those. So the NIA and NCI jointly funded a study, ASPRI, starting in 70-year-olds, which increased death, not decreased death. So, this is how hard it is to translate animal rodent experiments into human small molecule trials. I just don't think we're going to make any progress in this without a basic biologic understanding and a pharmacodynamic marker. You have to know what the drug does, and you have to show it can do it, and then you can do a trial. And everything else up until then is kind of GNC type science, and that's not a good thing.
Great. Any additional questions? Well, Dr. Sharpless, I want to thank you for not only your contribution today to the seminar, but actually your contribution to the government. I think one of the things we get kind of jaded in looking at thinking about government, but you look at people like Ned, and you look at the contribution that they're making every day in so many parts of our government, and it becomes very easy to criticize our government from the perspective of the right or the perspective of the left. But you know what? There's a heck of a lot of people like Dr. Sharpless who are making an enormous difference to improve the human condition every day in our government. And Ned, thank you for everything you're doing. It makes an enormous difference to all of us. Thank you.
Well, thank you for the opportunity to come. I really enjoyed it. I wish this could be in person sometime soon, but a great topic to be addressing, and I commend you on a wonderful seminar.
Great. Thanks, Ned.
Thank you, Dr. Sharpless. So now it is my unfortunate pleasure to follow the talk. And I'll ask Leon if she can bring it up for me, please. I'm going to switch gears here and talk about a research opportunity using the Life and Longevity After Cancer Cohort, which is a cancer survivorship study embedded within the Women's Health Initiative. My overall goal is to emphasize some of the gaps in our knowledge of cancer and aging at a population level and invite you to consider using WHI and LILAC to help address them. From an epidemiologic perspective, age is the most consistent cancer risk factor, having strong associations across many cancer types. More than half of all cancers are diagnosed in people over the age of 65. Yet epidemiologic analyses typically consider age as a factor to be controlled for, not to be examined further. We don't think of age as the driving force per se. It's not the number of candles you've blown out at any point, but rather the total smoke you've inhaled that matters. So age serves as a surrogate for cumulative exposure to factors that are of not interest in a particular analysis. We use it in a crude way to control for the lifetime implications of lifestyle and environmental factors that we haven't or couldn't measure directly. Using age in an analysis like this may capture some of the biological processes and changes that come with advancing age. Hormonal factors, genomic instability, inflammation, senescence, immune function, variety of things. Rarely do we measure all of these in the same organism to understand the cumulative effects over time and across these mechanisms. But of interest to us here now is whether these biomarkers, either individually or jointly, could more precisely reflect an organism's biologic age in a way that better predicts cancer risk and resiliency than chronic age does, chronologic age. Of course, cancer, and particularly its treatment, may affect these very same biologic processes. In fact, some therapies directly target these mechanisms. There are also well-established side effects of treatment that result in conditions normally seen only in advanced age, such as heart failure. Importantly, cancer patients may experience frailty, sarcopenia, cachexia, cognitive effects, disruption in sensory function, and even social function, all of which occur to some extent in normal aging and may be related to these processes. But these aging conditions have a significant impact on patients' well-being and potential survival. The growing size of our survivor population, now referred to as the silver tsunami, makes the non-cancer effects of cancer therapy a more important consideration, particularly in the care of older patients. Nearly 2/3 of the 16 million U.S. cancer survivors in 2016 were over the age of 65, and this fraction is rapidly growing. Their health concerns, including the ability to live independently without treatment-imposed deficits, becomes a priority in treatment decisions for both patients, providers, and their caregivers. There are large research gaps regarding cancer and aging, many arising because we simply haven't collected the data. While clinical trials now typically avoid exclusions based on age alone, still their enrollees skew towards younger populations based on other aging-related criteria. In addition, follow-up of clinical trials is usually limited to 5 years, and the data collection is understandably confined to the specific cancer outcomes and regulatory required adverse effect reporting. Aging-related measures simply fall out of scope of these trials. Finally, most epidemiologic studies of cancer have cancer incidence or mortality as the outcome. Historically, they haven't captured treatment information or other age-related health outcomes, nor have they collected biospecimens needed to address some of these questions. Some of this is changing, but slowly, and the available national database is still quite limited. A National Academy of Medicine workshop was convened just a month ago that explored this issue in depth. How can we improve the evidence base for treating older patients who represent the largest segment of cancer patients? This was particularly targeted towards cancer clinical trials, and if you're interested in expanding access to clinical trials for geriatric patients, I highly recommend that you go back and view that workshop. Arthi Huria, a geriatric oncologist at the City of Hope, had a specific passion and vision for this work. A compassionate physician, a thoughtful and generous collaborator, and a brave and inspiring leader, she initiated the NCI-funded Cancer and Aging Research Group, led geriatric oncology work in the Alliance Cooperative Group, and was a leader in other professional organizations to promote geriatric oncology. My LILAC colleagues, Dr. Betty Kahn and Electra Pasquette, introduced me to her as we started to think about developing an aging focus for LILAC. Over a period of several months in 2018, Arthi immersed herself in the WHI and LILAC resource to learn what we had and what we needed to make a significant contribution to this field. She graciously contributed her time to developing and refining the scientific agenda for our renewal proposal and agreed to serve as a multiple PI. Our proposal was submitted for the November 5th, 2018 deadline, and less than 2 days later, she was tragically taken from us in a traffic accident. We continue to miss her leadership, but are very committed to trying to fulfill her vision for this effort. What attracted Arthi to LILAC and WHI? Well, WHI is a compelling place to study aging. Initiated back in the 1990s by Dr. Bernadine Healy, the director of the NIH at the time, this program was designed to address the major causes of morbidity and mortality in postmenopausal women. If you know only one thing about the WHI, you know of our clinical trials, and specifically our randomized trial of menopausal hormone therapy. This trial component was designed, in fact, to test an aging-related hypothesis, that aging leads to menopause, which results in so-called estrogen deficiency, which purportedly resulted in a higher risk for coronary heart disease. We hope to intervene in that mechanism by adding estrogen back in and thereby preventing coronary heart disease. And you probably know the rest. But in truth, all of the WHI trials were testing aging-related hypotheses, since these were designed to prevent cancer, cardiovascular disease, and osteoporotic fractures. At the outset, we as investigators were focused on the clinical trials testing these prevention strategies, but our interest was in getting a comprehensive assessment of their impact on older women's health, and that had the natural consequence of creating an infrastructure that could be used to address many important issues associated with aging. And the parallel observational study in over 93,000 women supported an even broader research agenda. So I want to give you a brief reminder of who this cohort is. The overall eligibility was to be generally healthy postmenopausal women, and these women were recruited at 40 academic medical centers around the U.S. They were well characterized in enrollment with all the usual risk factors needed for epidemiologic studies of chronic disease. We also conducted a brief physical exam in all 161,000 women and collected high-quality fasting blood products and DNA from everyone. More extensive assessments were done in specific subsamples designed to reduce cost. Follow-up for these women have been remarkable, with annual response rates exceeding 90% for the first 20 years and remaining above 80% to the present. We've documented outcomes for our target disease of cancer, cardiovascular disease, osteoporotic fractures, and mortality. We also collected self-reported events of many other age-related comorbidities, such as type 2 diabetes, hypertension, osteoarthritis, COPD. We've also linked our database to Medicare for those participants who are in Medicare Part A and Part B, dating back to 1991, and we've recently received the Medicare Part C data beginning in 2015. These data allow us to expand our database significantly to include information on medical procedures and treatments and their associated costs, expanding the analytic opportunities considerably. We also query the National Death Index regularly to obtain vital status for participants no longer in active follow-up. We update patient-reported outcomes such as health-related quality of life, activities of daily living, and some lifestyle and mental health conditions at specific time points. Annually during the clinical trial until 2005, and once during follow-up in the observational study, we repeated key elements of the baseline physical exam. The WHI database has also been supplemented with separately funded ancillary study data, usually limited to specific subsets. Examples of these of particular relevance are those of cognitive function and dementia, eye disease, sleep disturbance, frailty, sarcopenia, and physical activity. In 2013, we conducted a home visit in nearly 8,000 women from across the country to get more detailed aging information. During this visit, we repeated the key elements of the baseline physical exam, obtained objective measures of physical function, and collected a new fasting blood sample. We made a special attempt to involve older women and women of color in this effort. So you get the picture that the WHI database is a reasonably comprehensive assessment of these women's health at baseline and continues to be so as they age through the last 25 years. We still have over 60,000 women in active follow-up, currently between 72 and 103 years of age. But what about cancer? At this point, we've documented over 35,000 cancer diagnoses. Our rates run parallel to those seen in the SEER registry over the same time frame, suggesting that this cohort of volunteers, while clearly not a random sample, are generally reflective of the larger U.S. population in this age range. The big cancer burden, in terms of incidence, are the usual suspects: breast, lung, and colorectal cancers, but we've seen a significant number of others, including endometrial cancer, melanoma, non-Hodgkin's lymphoma, leukemia, and even ovarian cancer. Not surprisingly, cancer mortality is driven by lung cancer, as over half of these women reported a history of smoking, although only 10% were smoking at entry. Most of our breast cancers are early stage, so breast cancer mortality is relatively low. Because WHI is large enough to have observed many cancers, and we continue to follow these women after their diagnosis, WHI can start to bridge the gap between traditional epidemiologic studies and cancer clinical trials. But to do this, and particularly to study the interplay of cancer and aging, we needed a bit more information. In 2013, my colleagues, Betty and Electra, and I received a cancer cohort infrastructure grant to initiate the life and longevity after cancer study. This first cycle of LILAC was designed to augment the WHI with information needed for cancer survivorship research. We chose to focus on cancers for which WHI has sufficient numbers in itself to support reasonably powered studies, as well as those cancers for whom survivorship studies are relevant. So we selected the 8 cancers listed here. So we collected first-line treatment and recurrence through Medicare linkage where possible and through self-report and medical records collection otherwise. We also collected patient-reported outcomes via LILAC enrollment and follow-up questionnaires. Topics covered targeted the late and long-term effects of treatment, those areas that we thought would complement usual clinical trial databases. We also collected formalin-fixed, paraffin-embedded tumor tissue samples from a subset of solid tumors, which can be linked to the WHI repository of blood and DNA samples. Among these selected cancers, 48% of women met our eligibility criteria to approach for consent and questionnaire data. We had a high response rate that only seemed to dip in the oldest old. For feasibility reasons, we focused most of our work on cancers diagnosed in 2001 or later. In addition to those who have consented, we were able to collect treatment, clinical outcomes, and tumor tissue under a waiver of consent for those who had died prior to LILAC initiation. So thus, our expanded database for LILAC includes data for nearly 13,000 cancers. We have patient-reported outcomes for nearly 8,000, treatment data from either Medicare or chart review for over 11,000, and tissue samples for over 4,000. You can see the breakdown by tumor type in this table. Through LILAC, we want to understand the impact of cancer and treatment in important subgroups: women over the age of 75, and those with comorbidities and other aging conditions that may have excluded them from prior clinical trials. We want to help clinicians know what kind of aging assessments are useful for guiding care decisions. And we believe that the LILAC resource will be particularly valuable for assessing biomarkers of aging to inform oncologic research and practice. Marthy's particular interest was to use these data to address the specific impact of cancer on aging by using the framework suggested in this diagram. Assume we have some measure of biologic or functional aging, and the level of function is expressed on the vertical axis. We expect that at some point in mid to later life, this quantity will start to decline with time, as represented by the solid black line. In populations that don't have cancer or another major health event, this would be considered normal aging. At some point, the decline may reach a level that would be classified as a disability, as indicated by the dotted green line. So what happens with cancer? Our hypothesis is that there are acute effects of cancer, depicted here in the dotted or dashed blue lines, that may be rather variable by cancer, treatment, and person. But we expect some rather short-term drop in these measures, and that as depicted here. We also expect that most patients will recover from these effects, at least partially. The extent they return to what would have been their level of function absent the cancer could be considered a measure of their resilience. The next question is then, what is their prognosis for these important areas after the recovery? Does the decline with time parallel that we would expect of a normal person without cancer, what Arthi referred to as a phase shift hypothesis? Or is the decline more rapid? That is, does the cancer event or treatment accelerate aging? These questions have important implications for clinical practice. Knowledge of these trajectories and the ability to link them both to patient characteristics and treatment options could help us better tailor treatment to older populations. The population is not homogeneous, and chronologic age is not a good enough predictor of resiliency. We want to be able to better characterize these patients in ways that can guide therapy. So we are now at a point where LILAC data can be brought to bear on these issues. Our aims going forward are to assess aging trajectories where we can, or at least to compare true pre- and post-diagnosis changes for a range of functional and biologic variables. We want to compare these trajectories and changes to corresponding analyses in similar cancer-free women. And we also propose to complete a home visit following the Long Life Study protocol in 2,000 LILAC cancer survivors to provide an additional longitudinal database. We plan to promote the use of this resource broadly, and in the meantime, we're continuing to enroll and follow women eligible for LILAC. So the development of an age-related cancer-free control group seems critical to understanding the role of cancer in aging. We have developed the WHI data set is ideal for this purpose, giving us women who recruited from the same areas, who provided parallel data over the same period of time. So we've selected a cancer-free control group, matching up to 5 women who are free of cancer at the time of their matching case diagnosis. And we match these based on age and original WHI enrollment information. So this large cancer-free control group allows for precise estimates of aging trajectories in cancer-free older women. It will also serve to support the analyses of pre- and post-diagnosis changes compared to measures taken at the same time intervals in comparable controls. And for information only available in the post-diagnosis interval, we can make simple cross-sectional comparisons in cases and match controls. As a first step, we've completed preliminary analyses of aging trajectories as assessed by self-reported physical function in cancer-free women using 20 years of WHI data. These analyses suggest there are 4 general trajectories present. 2 groups start out at very good or excellent levels and decline slowly, virtually in parallel over these 2 decades. The 3rd group also starts at a very good level and stays there for most of the first 10 years, and then declines rather rapidly in the 2nd. The 4th group starts at a significantly lower level of function and experiences a rather consistent decline throughout follow-up. It will be no surprise that age is one of the factors that distinguishes the membership in these groups, but also important were body mass index, self-reported general health, physical activity, and reported falls at baseline. Our next question is, what do these trajectories look like in women who get cancer? So this analysis becomes more complex. WHI data and blood collections were timed according to our prevention trial protocol. So to look at pre- and post-diagnosis trends, we must realign the data for these cases and match controls around the time of diagnosis. The upper half of this graph, we show the data collection for a woman who is diagnosed early in her WHI period. So a woman like this would contribute very little information on the trajectories in the pre-diagnosis period, but would provide a lot of information on the trajectory in the post-diagnosis period. For a woman in this lower part, her cancer diagnosis comes late in her WHI experience. She would contribute a lot of information on pre-diagnosis trajectories, but we'd have little information on her cancer survivorship experience. The infrequency of some collections, particularly biospecimens, will limit their ability to address trajectories as just outlined, but we can conduct the pre- and post-diagnosis comparisons in women with cancer and compare them to those without cancer to provide evidence of the magnitude of changes in biomarkers of aging. In 2022, WHI will initiate another round of home visits in an estimated 4,200 women who participated in the first round, repeating these anthropometry and physical function measures, as well as conducting an additional blood draw. LILAC will support home visits with the same protocol to another 2,000 cancer survivors, providing an additional longitudinal data point on a post-treatment blood draw done in parallel to these cancer-free controls that can be used to augment these trajectory analyses. So this extensive WHI database can help fill the noticeable gap between studies of cancer incidence. So now an invitation. Our goal is to maximize the use of this resource, particularly for studies of cancer and aging. To catalyze this effort, we're issuing a call for concept proposals to use the resource. We're looking for collaborators with expertise, interest, and ideas for manuscripts or grant proposals that would effectively address these gaps. A manuscript proposal would simply describe an analysis of existing WHI or LILAC data. A grant proposal would propose to collect additional data, for example, through additional questionnaires, through enhancements to the home visit protocol, or through analyses of WHI or LILAC specimens. Priority will be given to proposals that are aimed to help us understand the interplay of cancer and aging, as we just described. But other scientifically meritorious proposals will be considered as our resources permit. We're asking for a 2-page concept proposal to be submitted by the end of March, and we will consider it with the help of our external advisory board and get back to you by the end of May. If you have any questions about this, you can study our WHI website, which has extensive information about the data that's been collected. Contact our help desk for more tailored questions, or contact any of the 3 PIs of LILAC listed here. Unfortunately, LILAC cannot offer any direct funding, but for the concepts selected, we'll provide analytic support as well as the assistance of LILAC investigators in the preparation of grant proposals. For those using LILAC tissue samples, we'll cover the cost of sample retrieval, but we can't, unfortunately, cover processing and assay costs. For WHI specimens here, blood, DNA, or urine, the grant proposal must include all the costs associated with retrieving, shipping, processing, and analyzing those samples. We plan to use any concept proposals that involve the home visit to finalize the selection of LILAC participants to be included. Aspects that we are considering include which cancer types to involve, as well as the current age of the participants, the time since diagnosis, the treatments they received, and aspects of health disparities and health equities. I want to draw your attention to a recent NOCI from NCI and NIA entitled Understanding the Effects of Cancer and Cancer Treatment on Aging Trajectories and Aging Outcomes. The purpose of this NOCI, jointly sponsored by the 2 agencies, is to solicit applications that aim to better understand the effects of cancer diagnosis and subsequent cancer treatment on aging trajectories and aging outcomes. The background statement here looks like something that Arthi had her hand in, and WHI and LILAC is well positioned to support the first 3 specific research objectives called out, in particular, investigation of aging trajectories associated with specific cancer treatments, and identification of cancer survivors at risk for an accelerated aging phenotype. examination of the effects of specific cancer treatments on aging biology that may alter aging trajectories or aging outcomes, and development and or validation of tools, technologies, measures, or techniques for the identification of late emerging effects and aging phenotypes among cancer survivors. In addition, WHI and LILAC are willing and able to support selected clinical trials in this population. So if you have ideas that can be implemented on a nationwide basis, we may be able to support that research objective 5 as well, which is the development and or testing of interventions designed to prevent, mitigate, or reverse the adverse aging-related effects of cancer and cancer treatments. Please reach out to us directly about this opportunity, however, because clinical trials have many additional considerations that we should discuss. Finally, I want to end by acknowledging my collaborators, Arthi Huria, Betty Kahn from Kaiser Permanente of Northern California, and Electra Pasquette from The Ohio State University. I also want to thank our terrific staff in LILAC, and particularly Soumya, who conducted the novel trajectory analyses with guidance from Dr. Joao Luo. This work wouldn't have been possible without the marvelous WHI participants and our WHI colleagues. We've also benefited from the support of our NCI program officers, first Joanna Lena and now Lisa Galicchio, who I would note is also the contact person for the NOCI I just described. Finally, the WHI is funded by the NHLBI, and we're still celebrating its renewal this month. And LILAC is funded by a generous grant from the NCI. Thank you so much. We're at the top of the hour, so I don't want to keep those who are around waiting. It is my great pleasure to introduce Dr. Supriya Gupta Mohili. She is the Philip and Marilyn Werheim Professor of Medicine and Surgery at the James Wilmot Cancer Institute at the University of Rochester. She's a board-certified geriatrician and oncologist. She completed her medical training at the University of Chicago, where she also received a master's degree in health outcomes research. Her fellowship was funded by ASCO and the John Hartford Foundation initiative to train oncologists in the care of older adults. Her research interests include the evaluation of patterns of care, health outcomes, and quality of life related to the treatment of systemic cancer in older patients. In 2013, she was awarded a PCORI grant and an NCI award to evaluate whether geriatric assessment can improve outcomes of older patients with cancer. Both of those studies have been completed, and she'll be talking about them today. She founded the Specialized Oncology Care and Research in Elders Geriatric Oncology Clinic at the University of Rochester and is an integral member of the NCORE research base there, where she leads the cancer care delivery research efforts. She's an expert in geriatric oncology with over 200 publications in the area. She served on the editorial board of JCO and is editor-in-chief of the Journal of Geriatric Oncology. She was the chair of the ASCO Geriatric Oncology Task Force and the ASCO Geriatric Oncology Clinical Guideline Panel, and received the B.J. Kennedy Award for Scientific Excellence in Geriatric Oncology in 2018. She's one of the co-leaders of the Cancer and Aging Research Group, where she's multiple PI of their grant, and she's a recipient of a K-24, where she fosters the careers of promising early career clinician scientists in geriatric oncology. So her credentials remind me of Arthi's, and I think she's one of Arthi's legacies. So Supriya, so glad to have you. Please.
Thank you. Well, it's really a pleasure to be here today, and I want to thank Garnett and all of the leadership of this effort for inviting me. And I know you've had some really impressive talks today. Arthi was one of my mentors, and we still are following in her footsteps. And I'll really talk a lot about what she loved, which was really improving care delivery and outcomes for older patients with cancer. My research, as Garnett described, is very T4 oriented. It's models of care delivery. And I think it's important to put what we've learned at the point of care into context when we're thinking about new research endeavors. Especially in thinking about biologic measures of frailty and sarcopenia and things that we think really can be predictive of outcome. when we're looking at those new translational markers, we want to make sure that we can use them in the clinic with clinicians, because ultimately that's really how we will improve care delivery. So I'll talk first about the ASCO geriatric oncology guidelines, which really summarize the state of the art for geriatric assessment as a measure for assessing health status in clinical oncology. And then, as was mentioned, we've conducted 2 large studies within the University of Rochester NCI Community Oncology Research Program that evaluate geriatric assessment. And we now have very high quality phase 3 data that shows that geriatric assessment improves outcomes of older adults with cancer. And then I'll end with talking about efforts to improve the evidence base. This was another love of Arthi's, and she worked across multiple stakeholders to improve clinical trial design, improve sort of assessment at the FDA level for registration trials that are putting forward treatments that would be used for older patients. And then also work that we're doing at the NCI to integrate geriatric assessment into clinical trials. So why is geriatric assessment important? Well, we know that older adults with cancer have other medical problems, and these are just not comorbidities. I like to think of them as disabilities or geriatric syndromes. And we know that comorbidities, disability, and geriatric syndromes are interrelated, but not the same thing. And each are independent predictors of outcome in older adults. So the common concerns I see in my geriatric oncology clinic are needing assistance with daily activities, having comorbidities, mild cognitive impairment, and sometimes even severe cognitive impairment, for which I'm diagnosing for the first time, despite having an older adult who's seen medical oncology, radiation oncology, surgical oncology, and assigned consent forms for chemotherapy. It's pretty common that I'm diagnosing cognitive impairment in patients who may lack capacity for decision-making. Limited social support, living alone, transportation issues, polypharmacy, and frailty. And unfortunately, patients with these issues are not participating or have not participated in registration and cooperative group trials, yet they want and deserve to be considered for treatment that could be safe and efficacious for them. This is one of my patients in my clinic we're Wilmot Cancer Institute, who had rectal cancer, and I struggled with finding evidence-based treatment strategies for this patient who had multiple of these conditions. And his daughter, a caregiver who is a nurse, also had cancer herself. And so we had to think of a comprehensive treatment plan for the 2 of them that would allow them both to be safe during treatment. A geriatric assessment can do this. So a geriatric assessment is an approach to the evaluation of the older patient, leading to early identification and treatment of areas of vulnerability. And I think of these constructs as aging-related domains. So function, objective physical performance, which is really mobility, comorbidities, cognition, nutritional status, psychological status, and social support. We know that in community-dwelling older adults, each of these domains are independently predict morbidity and mortality in older patients. And the guidelines that we wrote with ASCO also summarize data that show that each of these domains independently predict morbidity and mortality in patients with cancer. So here was the guideline. This was published in 2018, and ASCO does a really great job putting together guidelines. Their staff go to the literature, look for large trials as preference, but if trials do not exist, they'll look for large prospective observational studies. They put together the literature for the expert panel, which is listed here. Then the expert panel synthesizes the literature and rates the strength of the evidence and the quality of the evidence. Our expert panel was very diverse, with geriatricians, medical oncologists, community oncologists, oncologists in the cooperative groups, and nursing colleagues. And we were able to come together answer 4 questions and also develop a practical way that geriatric assessment can be implemented in research and in the clinic. So the ASCO geriatric oncology guidelines address 4 questions. Number one, should geriatric assessment be utilized in older adults with cancer to identify those at highest risk for adverse outcome from chemotherapy? We chose chemotherapy because that's where the data was the strongest. We know that geriatric assessment also helps identify patients before surgery who are at high risk from adverse outcomes. And we know less about the newer sort of therapies like immunotherapy and targeted agents. But we focused on chemotherapy for this guideline. For number two, if one was yes, which tools should clinicians use to predict adverse outcomes? Number three was, when we're making decisions for patients who are undergoing curative intent treatments, what kinds of prognostic models can we use to assess non-cancer specific life expectancy? The reason for this is we're often seeing older adults who are in their late 70s or 80s who have other medical problems that could affect life expectancy, so that a 6-month course of chemotherapy may not be in their best interest due to both risk of the treatment, but because they may not live long enough to benefit. And so we do need to assess with using life expectancy models. And then how can geriatric assessment be used to guide the management? So in terms of questions one and two, the data supported, and this was more than 60 well-designed studies, that yes, geriatric assessment should be used for all patients undergoing chemotherapy. The evidence quality was high and the strength of recommendation was strong. There was less data to support one tool over another, and therefore the evidence quality was moderate and the strength of recommendation was moderate. Having said that, the panel was able to put together a very practical way to implement geriatric assessment in the clinic. And this is designed for any oncologist, even community oncologists who are busy, because this probably takes 10 to 15 minutes, and staff other than the oncologists can implement a lot of these workflows to get the data for this, these tools. And so in summary, in this box, this was a minimum data set for older patients with cancer getting chemotherapy. If we're deciding upon chemotherapy and trying to assess chemotherapy risk, we should use a chemotherapy risk tool like the Cancer and Aging Research Group risk tool. We should assess life expectancy with ePrognosis. Both of these tools are online and are very quick to do, and many of the questions actually overlap between the two. A functional assessment, which is instrumental activities of daily living, which is, measures the ability of an older adult to live independently in the community, very predictive of outcome in all settings and very quick to do. Not a specific measure for comorbidity, but using our general way of assessing comorbidity in the clinic, since we do a decent job when we're assessing patients in oncology clinics. So medical record review would suffice for comorbidities. One question for falls. Falls are very predictive of outcome as a geriatric syndrome. Using a validated tool for depression, which is part of other ASCO guidelines. Screening for cognition, which happens rarely, but is a strong recommendation because of the lack of ability for clinicians, including myself, to identify cognitive impairment in a patient without a screening tool. And then screening for malnutrition using weight loss or body mass index. In terms of question 3, estimating non-cancer specific life expectancy, ePrognosis was designed by, ePrognosis has 2 tools developed by Dr. Schoenberg and Lee, and there's a combined model in there. And the evidence quality was high that it predicts mortality, but insufficient because we don't know how these calculators are used in the decision-making process. But again, very easy to do. If you're using it at the point of care, put no in for presence of cancer because we really want to assess non-cancer related life expectancy to be able to make decisions. And then lastly, in terms of guiding management, we brought in information from our geriatrics colleagues from the American Geriatric Society guidelines. Because really, when you think about it, an older adult with cancer is still an older adult. And many of these management interventions still apply when someone has an impairment on a geriatric assessment measure. And so for functional deficits or falls, we should employ things like physical therapy and occupational therapy, fall prevention. For comorbidities, involving the caregiver, talking to the primary care doctor, minimizing medications. If someone was to screen positive on a cognitive screen, assessing decision-making capacity, thinking about involving the healthcare proxy. Depression, thinking about a referral, involving social work, cognitive behavioral therapy, medication use if needed. And then weight loss involving our nutrition colleagues. So this is sort of a multi-pronged approach, multi-component interventions for caring for an older adult. When I was an early career faculty member, I was thinking about how to study a geriatric assessment model of care. And unfortunately, there wasn't data on individual interventions. And so I couldn't pull in gold standard interventions into a multi-component intervention. And thinking through how to develop a multi-component intervention was tough. And Arthi was one of my mentors for this, RO3, where we did a Delphi consensus of geriatric oncology experts nationally. These are experts who ran geriatric oncology clinics. And we asked them, what do you measure when you're doing geriatric assessment? And how do you implement care process options? We were able to develop consensus on certain things. One thing was all of the geriatric oncology experts believed that every domain was important. We asked, can we streamline it? Can we shorten it? And they said no. So all of the domains that I talked about before are still in our recommendation. In terms of assessment options, similar to the ASCO guidelines, multiple tests can be used, validated tests can be used to assess each domain. And so the test can be tailored to the practice setting, to the patient population. What's listed here on this line in the middle column are the ones that are most commonly used and have probably the best data for older adults with cancer. And then we asked about process outcomes. So if someone were to have an impairment on one of these tests, it could trigger these referrals and downstream interventions. And so for someone, for example, who's falling, we could implement physical therapy, occupational therapy, home safety eval, and this could be standardized into algorithms. And so that served as the basis for an intervention that we did multi-site in community oncology clinics. I'll talk about two studies. The first was PCORI funded. And for those of you who are familiar with PCORI, PCORI being the Patient-Centered Outcomes Research Institute, PCORI really values patient-centered outcomes. And we worked with patient and caregiver stakeholders to determine what the outcomes were for this study. What our patients told us is that communication was the most important thing to them. They wanted to be able to talk about what was worrying them with their oncologists. And many of the things that were worrying them, both patients and caregivers, were aging-related concerns. And they often felt like they didn't have space in the clinical encounter with oncologists to talk about aging-related issues that they were having, or to ask about whether treatment would have an impact on things they cared about, like their independence and their cognition. And so we picked two outcomes with them. One was direct communication about aging, meaning we audio recorded all of the clinical encounters with patients, oncologists, and their caregivers after having a geriatric assessment in the trial. And we also evaluated patient satisfaction with communication and caregiver satisfaction with communication. Our hypothesis was pretty simple, that providing a summary of the geriatric assessment results with recommendations for the interventions, like I described on the previous slides and in the ASCO guidelines, would improve communication about aging and institute the implementation of interventions. And so again, we did this in our ENCORE research base. University of Rochester is one of two ENCORE research bases in an academic center at a university. The other 5 are in the cooperative groups, coupled with NCTN cooperative funding on the therapeutic side. And so the PIs of the ENCORE research base right now are Dr. Karen Mustian and Dr. Gary Morrow, both of whom also serve as mentors to me. Our intervention was very simple. So we provided a summary and a checklist. And for this trial, I'll show you the checklist for communication. And this is, kind of hints at Dr. Atul Gawande's work that using checklists and providing checklists are very helpful. So when the geriatric assessment was done in the intervention arm, the oncologists got this checklist that they were told or asked to share with their patient, with the patients and the caregivers that were enrolled. They had complete autonomy to do whatever they wanted with it. They could throw it in the garbage. They could pick one thing. They could pick three things. They could do, we didn't dictate what they did. It's their patient, but we just provided the information. If a patient, so for this slide, if a patient screened positive on a cognitive screening test for impairment, the oncologist would be asked, did you discuss the patient's concerns about cognition? Did you elicit input and perspectives from the caregiver? Did you assess decision-making capacity? Did you elicit healthcare proxy information? And did you carefully weigh the risks and benefits of treatment given limited data for therapeutic treatment in older patients who have cognitive impairment? Our study design was cluster randomized, both the trials that I will talk about. And so we randomized community oncology practices. This was not a patient level randomization. And for the sites that were randomized to the intervention arm, for the oncologists that enrolled, for the patients that enrolled, for the oncologists that were enrolled, they got a geriatric assessment summary and a list of the interventions. And then we audio recorded the clinical encounters, and we assessed patient satisfaction and caregiver satisfaction, as well as a whole host of quality of life and functional outcomes. And arm 2 was just usual care. All patients got the geriatric assessment, but only alerts were given to the oncologist if there was significant cognitive impairment or depression, without really a robust summary or checklist. Our patients were all 70 and over with a diagnosis of advanced cancer or lymphoma. And we chose advanced cancer because there's limited evidence of, for some of these treatments in these more frail older adults, and very small therapeutic indices. So we wanted to see if geriatric assessment could actually improve sort of acknowledgement of risks and benefits. Having said that, all of our patients had at least one geriatric assessment domain impairment outside of polypharmacy. So these are frail older adults that were enrolling onto this trial, not the ones that are usually included on therapeutic trials. And we did that for a reason. All of these patients were receiving cancer treatment, but were not the kinds of patients that were included in clinical trials that established the safety and efficacy of treatment for those treatments. We also enrolled one caregiver, and the patient got to choose the caregiver. The definition of caregiver here was developed by our scoreboard patient and caregiver advocate group, but patients who did not have caregivers were still able to enroll. And I hear quite frequently that enrolling older adults onto trials is very difficult. We didn't have as much problems because we partnered with our patient and caregiver advisory board. And because I believe this research question was very important to community oncology practices and the patients and caregivers that they serve. So we were able to enroll 305 oncologists, 541 patients, and 414 caregivers within the PQuarry funding period. And I'll now go to results for this PQuarry trial, but I'll do it by participants. I'll start with the oncologists. The oncologists were enrolled onto the study. They answered a survey at the beginning, and we asked them questions about their perceptions of medical care for older adults with cancer. And they told 90% agreed that the medical care for older adults with cancer needed to be improved. Yet only 23% were using geriatric assessment to help make decisions for patients in their practice. And oncologists, they communicated they wanted more training and they wanted guidance about how to do this. Going to the patients, I was quite surprised by the number of geriatric assessment domain impairments that the patients presented with. So we usually consider about 2 domain impairments as being frail. It correlates with frailty indices like Fried's Frailty Index. And what we found was the patients enrolled onto this trial, and yes, we were selecting the more frail patients, but the median number of domain impairments was 4.5, which means that patients not just have one domain impairment, but multiple. 93% of them were impaired with physical performance, 25% were impaired with psychological status, and more than a third were impaired with cognition. And these were all patients receiving cancer treatment. We found that geriatric assessment improved both patient and caregiver satisfaction with communication. And this was a low touch, one-time intervention where they got one summary after the geriatric assessment was done. It was generated through a web-based tool that the staff completed. But the persistence of satisfaction lasted 3 months, through our 3-month time point. So patients really and caregivers really valued this intervention. And when we looked at the conversations between oncologists, caregivers, and patients, we found that there were more aging-related concerns discussed in the intervention arm, and these conversations had a higher quality and led to more interventions. So this improved communication about aging and led to more aging-relevant care processes. So I mentioned before Scoreboard. Here's Scoreboard, a few of the members of Scoreboard. And Scoreboard really helps me now. I meet with them for 1.5 hours every month. It's a key core resource for the Cancer and Aging Research Group. And we published our work with Scoreboard. Dr. Gilmore did, who's a mentee of mine in cancer. And Scoreboard really helped me figure out what the next steps were. So we all have data from our studies, and yes, I had to focus on the primary aims of the study, but sort of secondary and tertiary aims, I didn't know where to focus. And Scoreboard really helped me because they told me, well, caregivers are really important to look at. Social support is really important to look at. So those are the results I'll talk about next, because that's the ones I focused on next. And here are some lovely quotes from Scoreboard talking about how they really value communication with the research team. So we did look at our caregiver health status. And what we found, which was not surprising, was that 75% of caregivers for older adults with advanced cancer are also older themselves, often spouses or friends. The rest are usually children. And the older caregivers had significant comorbidities and their own sort of physical health needs, and yet they were caring for their older patient who had lots of geriatric assessment problems and advanced cancer receiving treatment. What we found was that not only was geriatric assessment domains a predictor of poor outcome for patients with cancer, they're a predictor of poor outcomes for older caregivers too. And so we found that the number of geriatric assessment domain impairments was associated with poor quality of life, anxiety, depression, distress in caregivers. So this is an opportunity for geriatric assessment to be also used as an intervention for caregivers. And it's certain that in my clinic, when I do a geriatric assessment and I see many domain impairments, I make sure to turn to the caregiver and ask the caregiver how he or she is doing. Because I know that it's tough and the caregiver is a part of the team. And so we have to shift attention sometimes to the caregiver to make sure that the patient is also cared for and the caregiver is cared for in the ways that they need. There were lots of missed opportunities in the qualitative work that we did. Here's an example of a excerpt from a from the usual care arm from one of the clinic visits, where the caregiver says, well, she's been spending a lot of time in bed lately, and for some reason, she's not able to stand for more than a few minutes at a time. And the oncologist says, I see. And the caregiver says, well, she doesn't walk long distances. That's why she's in a wheelchair now. And the patient says, but I want to get out of this wheelchair and come in here dancing the next time. And the oncologist says, well, the hope is that we can help you feel better. How often are you taking Norco, a pain medicine? And so I see this as missed opportunities where the caregiver is really communicating distress. Caregivers don't tell the patient's oncologist, well, I'm distressed, please help me. They communicate about the patient, and they're really communicating the functional limitations that the patient has right now and how it's sort of a rapid decline. And the patient is communicating probably what's an unrealistic preference and goal. And so this could have provided an opportunity to really discuss goals and what's realistic and what's not realistic, and also support for the caregiver. And yet the oncologist sort of shifted the conversation to the pain medicine. And this is what geriatric assessment really helps with. It really helps sort of identify these missed opportunities and clarify goals. So those were the results for the PCORI study. I presented this second trial, which is a separate trial, but uses the same intervention at ASCO 2020. And this was an R01 funded study that looked at clinical outcomes related to geriatric assessment. And so lots of similarities with the previous trial in that it's cluster randomized and the intervention is the same, but the outcomes are different in that we were looking at grade 3 to 5 toxicity as evaluated by standard NCI common terminology criteria for adverse events. And we were looking at whether geriatric assessment could reduce toxicity without compromising survival. And a mechanism that we were interested in was influencing treatment decisions versus the interventions. And so again, the study design is cluster randomized, low touch, one-time intervention, where the oncologist got the summary and a list of the recommendations. And the patient population was very similar, 70 and over, at least one geriatric assessment domain impairment other than polypharmacy. But in this trial, we timed it so that everyone who enrolled was starting a new treatment regimen. They weren't completely chemotherapy naive, but they were starting a new regimen of using at least one chemotherapy agent or a regimen that had high risk of toxicity. And again, I was sort of very impressed with some of the study results. And here's another slide that sort of impressed me. We were able to enroll 718 patients onto this trial. And I was impressed by the sort of prevalence of adverse outcomes that happened very quickly after older adults started new regimens. So significant toxicity within 3 months, 61%. Half of them had a dose modification within 3 months due to toxicity. 17% discontinued treatment within 3 months. 15% entered hospice into 3 months, and 26% died within 6 months. And so this sort of raises the question of the treatment decision piece is, there's lack of treatment data for this population, and we struggle with treatment decisions, and we sort of often will err towards treatment rather than not doing treatment because we're uncertain. And yet patients get treatments that are probably too high risk and do poorly very quickly after starting these new regimens. We found that the geriatric assessment improved toxicity with an adjusted risk ratio of 0.74, and this was very significant. So in the usual care arm, the prevalence of having at least one grade 3 to 5 toxicity was about 71%. And in the intervention arm, that was 50%. And mostly this was evident in the non-hematologic toxicities. So these are toxicities like fatigue and diarrhea that really hurt kind of an older patient's functional status. We are also working with it within a UO1 in a tolerability consortium with the NCI. So we were able to apply for funding to use data from this trial to inform the tolerability consortium. And we're one of 4 teams. And our team is the one that has the data for older adults. One of the things that we did was work with Dr. Sandy Mitchell at the NCI to include pro-CTCAE, which is patient-reported version of the common terminology criteria for adverse events. And we utilized methods that were developed in this consortium to look at whether geriatric assessment improved patient-reported outcomes. And we found that it was true that if you looked at 11 core symptoms and even a sort of a host of patient-reported outcomes, the geriatric assessment also improved sort of the symptoms that the patients reported, not just clinician-rated adverse events. And here's the slide on mechanism. We were able to look at treatment intensity. And what happened was that in the intervention arm, the oncologist really used a start low and go slow model, which is what we advocate for in geriatrics. Because of limited data, there isn't like dosing that we know is absolutely the right dosing for older, more frail adults. And so in an advanced cancer setting, starting a little bit more carefully with a lower treatment dosing may be better, and then only escalating if the patient tolerates it, or keeping it stable. And so what we found was that more patients in the intervention arm received reduced dose intensity, but more patients in the usual arm had a dose modification after toxicity. And so for older adults, do we really want them to have lots of toxicities and then change the dosing, or do we want to be more careful from the beginning? And that's what geriatric assessment allows. We also found that survival was not compromised. So there was equivalent survival at 6 months and at 1 year after this low touch one-time intervention. So pretty powerful results for such a modest intervention. There was 4 trials that were presented at ASCO 2020, and here they're sort of all listed. Arthes started a trial called the GAIN trial that was presented by her mentee, Dan Lee, and it was completed by Dr. William Dale at City of Hope, that showed similar findings in a population of patients with both advanced cancer and curative intent treatment, that geriatric assessment reduced chemotherapy toxicity. And other trials, one from Australia, showed that geriatric assessment improved quality of life for patients receiving treatment. And then a pre-surgical trial showed that it decreased hospital stay. So now we have high quality phase 3 data showing geriatric assessment improves clinical outcomes. So I'll end this with just a few slides, kind of moving to the future. So where is the future? Well, the community has really been working towards improving clinical trial design and accrual for a long time, since 2013, when an Institute of Medicine report was published, and just last month when there was a National Academies of Science workshop about eligibility and treatment tolerability in older adults. FDA and ASCO have worked pretty closely with now the National Academies. And there's lots of work being done. And Dr. Kleppen has, from the Cancer and Aging Research Group, has done a lot of work partnering with leaders in the field. I don't think this has changed. This was the original IOM report from 2013 that says our cancer care system is in crisis because 60% of our cancer survivors and our cancer patients are older. The majority of cancer deaths are in older individuals. They have lots of other problems besides cancer. Very few people know how to take care of older adults because they don't have geriatrics training, and supportive caregivers are lacking. And here was a commentary that was published in JNCI from leaders that led an FDA ASCO workshop about improving the evidence-based, that the action items were to increase enrollment of older adults on trials, collect more information, including geriatric assessment related variables from treatment trials, expand real world data, and strengthen the collaboration with stakeholders to develop advocacy and policy solutions. More recently, Dr. Singh, who is our point person and our colleague from the FDA, helped to guide this inclusion of older adults in clinical trials guidance for industry, which kind of supports the JNCI statement. There isn't a sort of mandate yet, but there is a lot of sort of review happening to try to improve accrual to clinical trials. This being more focused on pharma registration trials, and the Cancer and Aging Research Group. So for those of you that aren't aware, the Cancer and Aging Research Group is funded by an NIA R33 grant, and there's now available cores to help guide geriatric assessment use in studies. And it's really a mentorship opportunity, so anyone can join, go on the website and apply for membership. There's no cost. You'll be invited to the meetings, and we have now NIH and FDA and lots of representation on those calls to help us design better studies. The last meeting that I saw Arthe at was this one. This was in October of 2018, where we had our first R21R33 meeting for the infrastructure grant. There she is in the middle. And our goals are to stimulate high quality research, such as what you're doing at Fred Hutch through these efforts, attract and mentor investigators, combine aging and cancer research in cores, and disseminate to the broader community. Dr. Kleppen now is our third MPI with me and Dr. Dale, and we work with lots of people nationally to help design trials. It's an open resource for anyone. And the Cancer and Aging Research Group has really partnered now with the NCI through a Cancer Moonshot initiative to really think about accrual of older adults to clinical trials. And these are NCI-funded trials through NCORE or NCTN. And there's now 3 kind of robust groups with, that are working on study design, accrual, developing infrastructure for clinical trials, assessment patterns, assessment approaches, and stakeholder engagement. And these working groups, the results and recommendations will culminate at a meeting in April with the NCI, and then will be disseminated to the cooperative groups and other stakeholders after that. And so the hope is to energize NCI into thinking through the best ways to evaluate older adults for studies. And then my last slide is I typically do honor Arthi because she was a driving force in all of what we're doing, the Cancer and Aging Research Group, and me personally as an investigator. And we do feel very strongly about carrying forward her legacy. And she said, even as we brace new exciting drugs and technologies, the time-honored medical tradition of compassion and active listening is the core of what we do. And so I'll echo that by saying that any research that we do has to translate to benefits for patients and caregivers, or what's the point? And so that's really what Arthi stood for. I'll thank my team and my funders. And if there are time for questions, I'm here. I'm done.
Thank you so much, Supriya. I want to close your slide or
Sure.
stop sharing so we can see you a little bit. And
Yeah.
happy to take questions. And maybe I will start off with one anyway. When you're thinking about implementing geriatric assessment in the clinic, do you think of starting it at a certain age of your patient, or do you look for some signs or symptoms that suggest that it needs to be done?
So that's a great question, and it's been discussed for many years. And I don't know that there's one answer. And often it's dependent on resources. So we at the University of Rochester have a consult clinic, and we're pretty busy. We see 20 to 25 referrals a week. We have 6 geriatric oncologists. We have the largest geriatric oncology program in the country. So these are duly trained geriatric oncologists, and each of them has their sort of own research interest. And we focus their research interests on a domain. So I have one early career faculty member interested in cognition, one interested in physical performance, one interested in polypharmacy, and they develop then the interventions for the clinic. So it's a nice way to do it. We don't have an age cutoff other than as a geriatrician. I can bill for patients age 65 and over, even when they've already seen one of my colleagues in the practice, because I'm billing as a geriatrician. So that's the reason it's a sort of arbitrary Medicare rule. When we asked our Delphi geriatric oncology experts, they recommended everyone over the age of 70. The data in the ASCO guidelines still suggests that there's benefit for everyone age 65 and over. If there's limited resources, there's a screening test that can be used called G8, which is a very quick screening test that we built into Epic. That can be used, and if someone scores 14 and above, those patients can go to the geriatric assessment. So there's multiple approaches, but still it's 65 and over because of the way that we consider geriatrics in the United States.
Great. So Betty has a question. Do you want to ask yourself, Betty?
Sure. So I was wondering, I guess you answered it. I was wondering, once you do the geriatric assessment, what are the modifications that the clinicians make? And you said immediate dose reduction, but are there other things? I mean, so it's up to each individual oncologist to make that decision. But if you had to recommend, if you were designing an intervention trial now where you actually dictated the intervention, what would that be?
So that's a great question. So I'm not sure that we can. So there's 2 components. One is the treatment decision, cancer treatment decision, if someone is at that decision-making point for treatment. And then the other is sort of general recommendations for older adults, management of older adults and frailty. And in our studies, we didn't dictate because we didn't know what the resources were. And we wanted the oncologists to prioritize what they thought was the most important. We now have an R01 to look at survivors. So these are older adults finishing treatment. And we dictate a little bit more the management, but what we're doing is having the clinicians, who in this case are the advanced practice practitioners, not oncologists, work directly with the patients to prioritize the top 3 interventions. And so we're not saying you have to do these 5, we're saying here's a list, and you should pick with the patient what they feel is the most important to them. And that way we think adherence will be higher. It will come into play resources that the practices have. These are very pragmatic studies because one clinic could have a social worker and another clinic won't. And so we can't really dictate exactly what they do, although in my mind, all of the management recommendations are important and are based on the evidence from the American Geriatric Society.
I guess, Supriya, that I was thinking a little bit more like, do we think that an 80% RDI that starts an immediate reduction is different than an 80% RDI that proceeds with toxicities and then has to be dose reduced? That's where you were kind of going with this, right? That it's kind of like, if you use this geriatric assessment, you're going to make some decisions that you might immediately dose reduce. And do we think that that's better than waiting to see if a person develops toxicities and coming out with that same RDI?
Yeah, so the RDI could be similar and the survival similar, but what we saw was the patient-reported symptom toxicity burden was less. The clinician-related symptom toxicity burden was less in the patients that got upfront geriatric assessment. Okay. So that is a meaningful outcome to patients. And we might not change survival, but we've been able to reduce, symptomatic toxicities. And I do think it matters also for clinical trial design. So this is what we're working on with the NCI. There's this sort of question about older adults and accrual is, should there be clinical trials designed specifically for older adults? And geriatric oncologists think yes, because for those vulnerable and frail older adults, they won't be enrolled on the phase 3 therapeutic trials that are enrolling all comers. The fit ones maybe will be enrolled, but the vulnerable and frail ones, like the ones in my studies, will not be enrolled. And so we need specific trials for that population, which makes up about 2/3 of older adults. Fit older adults is about 1/3 of the population. 2/3 of them are not fit. And then what you've recommended is, yes, we should be studying 25% dose reduction options for those phase 3 trials that are for older adults that are vulnerable and frail, and use geriatric assessment to select patients into those trials. And I think the NCI is interested in moving forward. There are some nice examples in the cooperative groups, one by Dr. Efrat Dotan for patients with advanced pancreatic cancer, where they're selecting in patients who are vulnerable using geriatric assessment and then using a tailored therapeutic approach, randomizing them. So I do think that is going to be the wave of the future, hopefully, if things work out, because what's happening now is everyone's just guessing, right? You don't know what to do and just guess and do whatever. Some people get full treatment because that's what it says to do, and then they get sick, patients get sick. Some people get dose-reduced treatment, even though they're fit, because nobody's doing geriatric assessment. So there's so much treatment decision-making variability.
So Supriya, there's been some chat in the motivated by Ned's talk to start with, but about nutrition. And nutrition's an element of geriatric assessment, but did I catch that it's really aimed at unintentional weight loss only?
So for this advanced cancer population receiving chemotherapy, yes, but you are correct that we are faced with sarcopenia and obesity in our population. So I think it's going to be in our survivorship trial that we're just designing now, just got thankfully approved by the NCI, now going to CIRB. We do both spectrums. So we're talking about healthy aging and living for the survivorship population and people who lost weight during there. So you've got this 2-pronged approach, but absolutely it's important for survivors.
Thanks. Any other questions? Okay. Well, I
So
really,
Garnet,
yes.
Garnet?
Yes, go ahead.
If no one else wants to talk, I could elaborate a bit in the chat, which suggested that a big gap in our knowledge about aging and cancer concerns nutrition, especially total energy intake. One could even hypothesize that because the older population that we've been hearing about tend to have lower energy intake, that might help explain the drop off in some survival rates that Dr. Sharpless mentioned early in his talk. But more importantly, even though, as Dr. Sharpless mentioned, the animal data is compelling about energy restriction and reduction in many different chronic disease incidence and mortality risks, we don't have an adequate research program on this, either for incidence or mortality or survivorship. And that's primarily because energy intake is not at all well assessed from our traditional approaches, whether it's food frequencies, food records, or food recalls. They're almost useless. So it may be among the biggest gaps in our knowledge that is not well recognized even by the broader nutrition community, that we need to find ways to address for both survivorship issues and for incidence issues. So I just like to get that topic out there. It goes from one decade to the next, unaddressed and unappreciated. It's one of the main leads we have for reduction in incidence and mortality, and we need to have better human data on the topic.
I'll echo that. We have an early career investigator in our research base, Dr. Amber Kleckner, who is thinking about this. I think it's hard though. We definitely need better measures, but it's a very behavioral thing for patients with cancer to follow a very specific diet or restrict calories. And so part of it is learning about the calorie restriction, but the other part is how do you get people to do it if that's what ends up being beneficial. And so we do want to learn about it, but if people won't do it, that it's going to be less translatable. And so both of those will have to happen at the same time.
So
Can I give a plug in for later this week, Ross? There is a great interest now in looking at energy restriction mimicking diets at the time of chemotherapy and seeing if it'll enhance outcomes. And we've got a talk later on Wednesday where someone's going to talk about energy restriction mimic diet as an adjuvant to hormone therapy, to tamoxifen. I think it's tamoxifen. And so she's got some interesting results. So it's not exactly what you were talking about in long-term reduction of mortality and incidence of disease, but there's quite an active field now in looking at energy restriction at the time of treatment and seeing how that improves the effects of treatment and at least short-term survival and recurrence. So
thanks, Betty. That's good to see that developing, and I think we do need some comparable effort on the incidence, the risk side, which is where the data are particularly lacking.
Yeah. Okay. So thank you, Supriya. That was wonderful talk. Very, very informative. Helps us understand a little bit what you face in the clinic and how there are real opportunities to improve care. And so thank you for that. So I want to remind you, we start again tomorrow morning at 9 o'clock. Tomorrow's theme is more on the biology. We have several strong speakers who will talk about different aspects of biological mechanisms associated with cancer and aging. And so we look forward to seeing you there. Same link. So have a great rest of the day, and we'll see you tomorrow morning.